my name is Robert Masson. I'm with Yuma County Agricultural Extension Department. It's a part of the University of Arizona that's designed to extend the resources of the university out into the community. So, oh my goodness, how fragrant. Here's a real star of the show. So I'll be introducing our speakers today. Without further ado, I'm going to introduce our first speaker, Dr. Bindu Padel, who's leading up our pilot program for hemp here. And she's an extension pathologist, so she's got a lot of background with diagnosing diseases here in Yuma. And she's gaining a lot of knowledge with hemp and how to grow it in a way that best fits our agricultural system.
Good morning, everyone. My name is Bindu Padel, and I'm the extension plant pathologist with the Yuma County Cooperative Extension. Just a little bit about myself. I was born and raised in Nepal. I got my bachelor's degree in agriculture science, majoring in plant pathology, master's, again majoring in plant pathology, more on the molecular side, plant virology. Plant virology again for my PhD, and then for my postdoc, I did some extension work and worked more on viruses, but I was more drawn to the extension part. So here I am at the University of Arizona. My program, I know you're all here to listen about hemp, but you're going to have to listen a little bit about plant pathology. The plant pathology program we have here, we do research and extension, and then plant disease diagnostic, and now we're getting a little bit into the hemp research. And one of the major components of our program is the plant disease diagnostic. So there's the National Plant Diagnostic Network, which is a network all around the country that has like about 200 labs, and we're one of them. Anything about the program, the plant pathology program, it will be available in our website. I'm using this platform to announce our next big event in terms of plant pathology. We're going to have the second annual field day at the clinic. It's a plant disease symposium and barbecue. Okay, is it better now?
Yes.
Okay. So on May 18, we're going to have the plant disease symposium. We have the whole Salinas driving down to Yuma, all 3 research scientists from USDA Salinas. Jim Correll is the distinguished professor in University of Arkansas, and he's been doing a lot of work on downy mildew and spinach in Yuma, so he's going to present about all the work and the information that he has generated in our area. And of course, your favorite plant pathologist. Alex and I have not decided what we're going to talk, but Stephanie is going to talk about what she does work in terms of fusarium wilt. They have done a lot of work, and she's going to present about it. On the industry work front, Dr. Ma will be representing Bayer Crop Science, and we'll have the representative from Green Goose Seeds that they have developed a fusarium tolerant cultivar on lettuce. So this is your opportunity to come and talk with them and ask them all the questions that you have in terms of fusarium wilt. Like Robert said before, any update, this is the IPM update. You wanted to include it in the newsletter, you email to one of those, or just email me and I can ask Marco to add you. So what happens with this is all the updates that you are getting is what's happening in Yuma area. So in terms of disease, in terms of insects, in terms of weed diagnosis and management, so this is all the update, any events, it all goes there. So now let's talk about hemp. How many of you are growing hemp? How many of you are thinking about growing hemp? How many of you have Kush? So I'm going to explain this on behalf of everyone in the university that I get a lot of emails, texts that, do you have stuff to sell? Do you want to buy? I want Kush. We're not involved in buying, or we're not involved in selling. All we do is research, and we absolutely got no Kush. So let's look at the history of hemp in Arizona in 2019. So around end of May, the Department of Agriculture started giving permission for growers to grow. And then by June, plants were already out in the field, and it was 120 degrees up there. Plants were fried, but we've waited for almost 70 years. We're not going to wait for another 7 weeks. So we went out and planted it. At that time, we're still not allowed to work on hemp. Sometime around July, we're allowed to grow out in the field and help the farmers, and I was allowed to do the diagnosis of hemp disease, and at one point, almost half of the samples that we got in the clinic were hemp, and that's how I got involved in the hemp projects. On September 9, we had the hemp workshop, and on October 29, finally, the university had the permission to grow hemp. And of course, I planted in November 7. You see the pattern here? I'm just one with the community. I don't wait either. But we wanted to go ahead and plant it because we wanted to generate information that if it does not work, we're going to tell you guys do not plant in November. So that was the idea. If you're in my last presentation, I was whining about how my friends were taking pictures like this, and when I go into the field, it was like this. But I don't give up. So we got it. It was in December in Casa Grande, and in a growers field, the plants were like really nice. So let's look at the 2020 vision we have, and I'll give you a brief walkthrough of what we're doing. The plant pathology program, we're focusing on the best management practice, but we're working on the plant vigor, plant health, because that's what our program is about. We will be doing a statewide trial on the CBD hemp. We'll be doing some autoflower trial at the growers field and the research center. We are getting into the seed and fiber, hopefully, in next few months. And since I'm a pathologist, we are going to do some cover crop trial using the seed and fiber hemp to see how it changes the population or the pressure of fusarium in lettuce. So it has been shown to have a good effect. It is a biofumigant. It has shown some promising results in terms of nematodes. So we want to see what it does with the fusarium. Varietal trials, all of it goes to Robert. This is a neutral third-party testing. This is all the things that he's going to do. Vetting the varieties, recording side-by-side performance, matching the correct variety for the region, and of course you'll publish all the data for public to review, so you get to pick what you want. Any information that you need on the varietal trial, you contact Robert. I know that there's a lot of interest in the hemp in the native nation, so any interest in terms of that, we encourage you to go through Trent. I mean, it will eventually come to us, but we are more than willing to work, but Trent will always be a part of whatever happens in the native community. So please go through him. He needs to know that there is a high interest. Okay, so we have a lot of speakers after me who are gonna focus on the production part. So I will focus more on the disease part. One of the problems that has already been seen is the fusarium wilt, of course. It is a systemic infection. It can come from other plants. You can see the vascular discoloration. That's how the plant dies, because that's where the food is transported back and forth in the plant. And then your plant just succumbs to death. This is how it looks in the field. You cannot say for sure that it is only fusarium that's in there. It could be more than that, but this is one of the typical symptoms. And we have already seen in here stem canker caused by Phytophthora. Anytime you think you have Phytophthora, bring the samples to us and we can do the diagnosis for you. PTM. A lot of the problem that you can see in the crown of the plant is because the plant is having wet fit. So you do not want that in terms of this plant does not like wet fit. Powdery mildew, we have not seen much of that in our area, thankfully. But it has been more a problem in the southeast. Okay, and then you would see a lot of mosaic and distortion. And I'll talk a little bit more about it in the next slide. What are we going to do with that? And there will be different kinds of herbicide and chemical damage that you will see. So when in doubt, you have to scout, not just for the male plants. You have to scout the weeds. You have to pull the weeds out, because if you have the weed growing next to your weed, your plant is not going to go get bigger, especially if they're very close to each other, and you cannot use herbicides. So be proactive with pulling the weeds out. And any of the disease. You see this disease, you see this. Anytime you see a diseased plant, just get rid of it, because it is staying there acting as a source of inoculum for your healthy plants. And there's not much you're gonna get in terms of production from there. So you're just putting your plant, your healthier plant in risk. Okay, so in our trial, where you will go after the workshop, you will see a lot of those. So I am keeping it because I'm doing research. For you, I do not recommend keeping them because you're not gonna get much produce. Other thing is, some say it's genetic anomaly, some say it's viral, some say it's thyroid, but the reality is no one had time and resource to look what is actually in there. No one knows. But I'm growing them because I'm going to freeze the tissue, and I'm hoping that one day I'll have funding and I'll have money to look at it. So that it's easy to find what it is in there. You do the thing called next generation sequencing, which is a very high throughput sequencing, and you can find out what is in there. You can get sequence of all the organisms in there, but it is about $2,000 to $3,000 per sample. So, and there's no money in research. There's money in marketing. I see a lot of money in marketing. And then again, I cannot stress this enough, and you have probably heard in a lot of other seminars and workshops too. Anytime the plant has weight fit, you are gonna have a lot of disease. So it's basically a lot of opportunist pathogens that are already in the soil, but then you have high moisture, and there are cool temperature loving fungi, there are warm temperature loving fungi. You have water in there, you'll have one or the other. So whenever you bring the plant for plant diagnosis, you need to fill out this form. Some people like to just put their business card in there, which is cool actually. But these are the information about the host plant, the symptoms, the history. You really need to fill them out. You can get the form in the UMA Ag Center. You can get it online. You can get it in the Arizona Plant Diagnostic Network. Whether you are sending it to me or any other diagnostic lab, please read the information very carefully. Every lab has its own set of rules. Every lab has its own set of direction, how you receive the samples, how do they want it. So be careful about that. We will have the molecular diagnosis available starting May. Anything about insect, it has to go to Dr. Palumbo because I do not know anything. He gave me the list. I'm just going to read it through. So these are the things that he saw in the last year in fall. Beet armyworm, corn earworm, pale striped flea beetles, grasshoppers, Lirio leaf miners, lygus bug, which he says he has not seen any observable damage, unidentified plant bugs, tree-corn alfalfa, Mexican leaf hoppers, white flies. Whenever you see white flies, aphids, thrips, insects like that, you have to be careful about those because they transmit a lot of viruses, but at the same time, you cannot also not spray insecticide. So there's not much we can do here. And then he says some melon aphids in December. So anytime you have any insect problem, this is the guy you need to contact. He is the entomologist. And you need weed diagnosis and management. You talk to Barry Tickets. He is the weed specialist. Okay, so you all are waiting for the field day, so I'm going to walk you through the trial that we did. Do you have a question? Okay. So it is the pilot project that we did that you're going to see after the workshop. Our focus was looking at the planting day trial, so we want to see if November is going to work or not. So we went and planted it. We did try direct seeding and versus transplant. We had drip and furrow irrigation going in there. We have a plot that we have 10 tons of organic compost manure added in there. And then we're looking at the disease and pests, and we're also thinking about seed-borne virus screening. Okay, so as you know, we're focusing on the best management practice trial. It is located at the UMA Ag Center. November 7, the variety is Maverick by Chiazin. It's an auto-flowering variety. The maximum temperature from then until now was 91 degrees Fahrenheit, and the lowest one we had was 32 degrees Fahrenheit. Right now, we are at week 3 of the flowering cycle. Looking at the male plants, we have had, it is a 1 acre, we had about 25,000, we seeded about 25,000 seeds. I would say we had closer to 80% germination. We don't have the exact plant numbers yet. We're gonna have a team of a drone company called Farm Flight. They're gonna do the plant count for us. They will be on site today. Feel free to talk to them. They are actually also developing a software where the drone detects the male plants in there. So they will have a setup there. So please go and talk to them. And looking at the male plants, that's a really, really good feminization. So this is how the plants looked in December 10, like after a month. This is how they looked in December 26. And by January 15, they were big. And this is how they look now. So there are a lot of dilemmas, starting from some people like purple, some people like green. Is purple the desirable color for everyone? Yes? No? You don't care? Okay. So there are still some confusion, like do you want feminized seeds or non-feminized seeds? It all depends on what you want. You have to look at the pros and the cons. The feminized seeds are expensive. The non-feminized are, more affordable. From our very, very limited experience, we had a small varietal trial of which you can look at it, but you cannot get in there. Today, you can only go in the autoflowering field. But from the small experiment that we had, non-feminized was not really worth it. We had more than 50% male, and then they all started flowering. Sometime in December, we had those frost for 2 days, so they all started flowering after that. And it was holidays, all the stuff were gone. So, and I had like 300, 400 plants flowering in there. Thankfully, my son is pretty smart. I taught him how male plants look like, and then, he just pulled all of them out. But yeah, over like more than 60% plant, we had male plants. Clone versus seeds, it's up to you again. When you are getting clones, make sure that whoever you go get the clone from, listen to the direction that they give you about hardening, about the transplant socks, all of those information. Be familiar what you are getting into. Know your risk, know your rewards. Seeds, you can direct seed them if the soil temperature is right. So on our trial, and this is just from one trial, so you have to keep that in mind. We had better rates, better success with direct seeding because the soil temperature was perfect. And then also, that was our first ever time trying to make transplants, so we're not good at it. And then drip versus furrow irrigation, we did not see any significant difference. We actually saw better results in furrow irrigation, and we only watered it like 3 times. All of this information that it will be given, you will get the fact sheet when you go to the field. But anyways, we only use furrow irrigation like only 3 times throughout the whole crop period, and the plants are actually looking better in the furrow irrigation. I think because we had rain, and then they got the natural rain, and then during the dry period, they probably extended out more roots, so they are more vigorous than the one in the drip. But at the same time, not every agriculture land has the privilege of getting furrow irrigation, and with drip, it's easier to apply fertilizer. Whatever you want to add in the plants is easier with the drip. So it's up to you to decide what you want to go with.
Was water conservation a factor in that at all?
No, not in this trial, but Andrea wants to do something on she is the next speaker, so she wants to do some work on the water usage and water conservation part. So when you go to the field day, you'll get the fact sheet there, and then there's a farm flight set up there. There's the Adivina. I think they're going to take more of the plants that they have in here there. You will go to the 8th Street. You will get out from here. Once you get to the 8th Street, you'll look for signs for the parking, or just look for a taco truck. There's a taco truck waiting for you guys. So anything on the hemp, this is a link that you follow to any progress that we have made. It will be available in the website, any results, all of that. The next big thing we're having is the summit.
Sorry, can you go back? They were trying to write that down and take pictures.
Oh, okay. So you have to do is you can Google just UMA plant pathology. When you Google UMA plant pathology, it'll take you to the plant health clinic, the hemp research, everything that's under the program. Just UMA plant pathology. Okay, so the next big thing on hemp is we're having the breakout session on hemp. It'll be me, and then this is the one that you really don't want to miss out on. Dr. Wellington from University of Florida, they have done a lot of work in terms of hemp in seed and fiber. So he has a lot of information that they have generated in the last 3 or 4 years, all the contacts, all the companies that you can talk to in terms of seed and fiber. So do not miss that one. You can miss mine. And then Dr. Matthew Breck will talk about the disease and pest management in industrial hemp. And then we're going to have a panel discussion where we have people from suppliers to like growers, and we'll even have extractors, and TSC consulting, all of them. Like we're going to have, you can have a panel discussion with all of them. So all the people in this here now, they're going to stay with me. However, we have some interns who have been volunteering in the hemp project from the beginning until now. So if you are looking to hire someone to work in hemp, they are ready to have a job which actually pays them. And they're around here today. So, so this project, as you know, there's not much funding in terms of research, and it has been possible because we have had help from so many people. So we literally started this project with no money. All we had was for the land to pay for the university. That was it. So, we would like to thank University of Florida, Clemson University, Kayazin for supplying the SIS, Morone Bioscience, Farm Flight, Keithley Williams, NutriNag, Rainforent, and Revolers Irrigation for all the feedback, all the help, all the resources that they provided us for this pilot program. And of course, Booth Machinery for allowing us to use the venue again, and Yuma Ag Center. And thank you to all the speakers and sponsors, Kaizen, Maroon Bio Innovation, and Edivina Nurseries. And whatever you see today in the field is because of the farm crew. So they've been working day and night. They've been working more than what we did. You know, they are attached to those plants. So thank you to our farm crew, and whenever if you see them, they should be in the field, just say thank you to them. And most importantly, the people who let me in their field. You know, like when the plants are not doing well, of course I have open invitation to go anywhere. But the growers, small, large, who let me in their field, knowing that I do not know anything about hemp yet, and they still let me in their field to see the difference in the healthy plant, non-healthy plant, to understand it. So when you let a researcher in your field, you're giving them just more than your time. You know, by looking at a plant, by looking at it, they can understand a lot more things than non-researchers do. So thank you for letting me in. Thank you for letting us in. And with that, if you have any more questions. Yes.
I know nothing about hemp, so I've got a couple of questions. So you've got boy plants and girl plants, right? Mm-hmm. Okay. At what age can you tell them apart?
I'm sure there are markers to tell them ahead of time, but with the research that we have, the males usually tend to develop faster than females. So you see those pollen sacs in the plant. That was back in my presentation. Let me see back if I can show you.
And what's your ratio of male to female, or do you know?
It depends. If it's not feminized seeds, you can have anywhere more than like 50% or even more. With a good feminization, you can have 5% or 1%, 2%. The one that we got from Kaizen, it was less than 1%. We only got 10 plants in 1 acre field. So we seeded like 25,000 seeds. So let me see, I'll show you the male plants.
Is hemp a perennial or an annual?
It's an annual.
Okay. So what type of growing season does it have? How many months?
I guess it depends. The full length dependent, you don't know. Auto-flowering varieties, after they get certain amount of light, certain hours of light, they go to flowering. Even auto-flowering, they're supposed to, most of them are short period from like 75 to 90 days when everything is ideal, but we planted them when it was still cold outside, so they are now November 7, so almost 3 months, and they're still growing. So this is how the male plants look like. So the female are more like the canopy is a little bit different from male. Male are more thin and lanky, and then they mature sexually before the female. So they have that pollen sac ready when the female starts flowering, and then that's when you pollinate. So you have to be extremely careful if you're producing for CBD. There's one. You have one plant. It's enough to pollinate your entire field. If you have seen a male plant and then you move it like this, you can see like billions of pollen streaming out.
So do you use bees for pollination or wind pollination?
I think wind pollination is gonna be just enough, especially for Arizona.
Thank you.
Thank you. Any more questions?
So the male version is better than the female, or is it vice versa? You want to keep the female or you want to keep the male?
We're keeping the females. So the unpollinated female flowers are the one that produce CBD. It depends. If you're producing for seed, of course you'll need the males. But if you're producing for CBD, you don't need the females. So whether you are going for feminized seeds or unfeminized, it also depends on your purpose of growing.
So for making canvas, what would you use the females?
For making what?
Canvas, and tints and stuff?
Oh, for the fiber?
Yeah.
I think you can grow either. Yeah. But there are some fiber varieties that are monoecious, not dioecious.
So for our next speaker here, we have Andrea Carter, who's going to talk to us a little bit about the native tribes and growing with them, as well as, it sounds like, some water conservation work. Is that right? Oh, wonderful. Yeah. All right, well, here you go.
Okay, so my name's Andrea Carter. Oh, thank you. Great. I'm a PhD student at the University of Arizona in plant science. I got my master's there and did my undergrad at Cornell in agriculture. So today I'll be talking about prospective research opportunities in industrial hemp and opportunities for Native nations. I myself am a member of the Powhatan Nation, which is in New Jersey. I'm from the East Coast, and that is why I'm interested in the opportunities of industrial hemp and to help Native nations economically. So I'll be talking a little bit about that. So just some background about my work. I've been working with Dr. Mike Ottman, who's a grain specialist at the University of Arizona, and I've been researching a low irrigation barley variety known as solar barley. And this was developed over 10 years of breeding for adaptation to low water conditions. So much of my work was field work, comparing conventional to these low water use barleys. And I was looking at rooting depth and penetration ability, as well as early stages of development. So we have the border cells and mucilage and root hairs. And the point of this was to see if there were traits associated with drought tolerance that we could use when breeding for adaptation to low water conditions for other crops. So the point of this, I share it, is to say that this type of work is what's needed in hemp. That we've done a lot of agronomic work in other crops, but we're really starting at ground zero with hemp. So this type of work of figuring out what helps plants grow and adapt to the Arizona climate can help us when we're breeding for Arizona adapted hemp varieties. So I also worked at a 200-acre organic farm, herb farm in Vermont, and I was post-harvest and production manager there, and they've been growing industrial hemp for the past 2 years and have their own line of products. So that's what really exposed me to the opportunities in hemp. But being out here, I thought, what can we do with the Arizona hemp business? So to start out with opportunities for Native nations, with the Farm Bill, it's allowed not only states but tribes to create their own hemp program. And so there are tribal hemp production plans. And you can view the approved plans online, and that can be a helpful resource for guiding your own creation as a tribe, how you want to plan it. They're all available. And this is really helpful for tribes having not only political sovereignty in how they want to design these programs, but also economic opportunity in how they design the licensing and how much they want to charge. So some tribes have done it where if it's a business primarily made of tribal members, they can waive or subsidize the licensing fees. So it can be a more inclusive industry in that way. Here's some nonprofits that are working with tribes to help them in their development plans. There's the Hempstead Project and the Intertribal Hemp Association. So those are worth checking out. And here's Winona LaDuke, the well-known activist in her own hemp field in the Ojibwe White Earth Reservation in Minnesota. And a lot of tribes are getting into this. As you can see, we have 25 tribal nations have already applied for, some of whom have been approved for their own hemp plan. And in your tribal plan, it will include how you plan to keep records, how you plan to give out licenses, how you will inspect, and how you will dispose of hemp if it is too hot. So you still have to meet the federal regulations, but there is some agency in how you design this program. And there's some helpful resources for designing these programs. So here's the contact information for folks at the USDA who will conference call and talk with tribal members and teams in their development process of their hemp production plan or their industrial plan. So they're quite helpful, as well as Trent Tigerstorm, and I myself am more than happy to talk to any tribes interested. I've been doing that in the past year, and it's quite exciting, and I'm really interested in it. So here's some opportunities that are also open to everyone. With the passing of the Farm Bill, now you can also qualify for whole farm revenue protection if you're a hemp farmer. This was not the case before. You can get crop disaster assistance. You also can be part of NRCS programs, their conservation programs, qualify for farm loans or beginner farmer programs. If you're a beginner farmer trying to get into hemp, you're no longer disqualified because you're growing that crop. And you can also now get organic certification for those of you interested. So this is pretty exciting because though hemp is a very regulated crop, it is still being considered a commodity crop with the same opportunities through the USDA. So going on to prospective research areas. Like I mentioned, there's so much to be done with hemp. We're really starting at a baseline. And so varieties, first off, Robert Mason is gearing up for these. As he mentioned, looking for companies to enter seed into variety trials. We also need to look at planting date. Duke Poly at the University of Arizona is planning a planting date trial in Maricopa, where he'll be planting from February to April. So that's useful, and we just need more of these and years of this research. Just like we have in barley and other crops, we need it in hemp. Best management, this is the work of Bindu, and she'll, as she mentioned, looking at compost, planting density, pest and disease control. All of these questions, or all of these topics bring about a lot of questions and a need for research in them. Another big thing is water use. So often hemp is touted as a low water use crop, but there's actually very little research to substantiate that, especially out here in Arizona or in the Southwest. So it might be a low water use crop in Oregon, where they're getting a lot of rainfall. You don't need to supplement and irrigate, but out here with the high evapotranspiration, is it really a low water use crop? And that's an economic question, is if you're putting all these irrigations on it, is it still economically worth growing? And lastly, the effect of heat and water stress on THC levels. This is a big issue, and perhaps why we saw plants going hot. So all of these research questions are important and will be the keys to success of people being able to grow this crop while remaining legally compliant. So it'll take advocacy and support from the industry, growers like yourself, and an expressed interest in these topics for this work to be done. And for that heat and water stress, the importance of that is exemplified by this story, and as Robert shared, a lot of crops going hot. So I don't think it's as dire as some of the news stories were making out, and Robert's numbers are a little more updated, but 35, I think is what he shared, % tested hot, and that was about 20 to 25% that had to be turned in. Now, that's the national average. According to the USDA, nationally 20% of hemp lots tested hot and had to be turned in. So it's not like Arizona's, we're going to rule it out or anything. But it does bring up the importance of heat and temperature stress. Or heat is temperature. Heat stress and water stress on this crop. And also the role of variable seed quality, as Robert mentioned, and genetic expression, where the seeds are coming from. If they're from Canada, how well are they going to do down here? And just the inherent variability in these crops and in hemp seed. Also, planting time and stress. So one reason why we might see high THC levels is that the cannabinoid precursor for both THC and CBD is the same one. So what we need to figure out is the processes and mechanisms within the plant of how these precursors are created and the management strategies to try and encourage CBD or at least minimize THC production. But this is tricky because the plant is producing the precursor and it's the shared one. So this is a study that looked at CBD and THC precursor content in plants over time. And what we're they looked at 3 different types. And what we're concerned with is the purple, which is the low THC or fiber type hemp. And what you can see is that the precursors of THCA and CBDA, these are the precursors to THC and CBD respectively, increased in the fiber hemp until the end of the season. They also increased when trichomes developed. So this is interesting because it would be helpful to have developmental stage signs of when you know that your CBD is high or your THC is high. And this is, we can't extrapolate, and I can't give you any like golden rules of when the trichomes turn a certain color, this is when you should harvest, but that's the type of work we could research and the type of topics we could look into to be able to have that type of developmental cue that would aid in knowing when it's the best time to harvest. They also found that THC and CBD increased in leaves and flowers during the last 6 weeks of flowering. So again, this is going to help us understand our harvest times. And these types of studies could be done and would really help growers succeed, I think, of really narrowing in when are we having low THC and optimal CBD levels. And this is important because the USDA knows that THC increases in the plant as it matures. And that's why they want you to sample your lot within 15 days of when you harvest. So here in Arizona, it's within 14 days of when you harvest, an agricultural agent has to come out and sample. And they want you to do it within that time frame because if you wait longer than that, let's say you sampled a month earlier, your THC levels will be dramatically different than when you actually harvest. So they want a clear idea of when that it's pretty close to your harvest levels. You cannot harvest until authorized by the department. An agent will come out and sample your field. They're going to test for total delta or just delta-9 THC at the moment. They will take a sample from the top inflorescence, the flowering portion of the plant, for up to 30 plants, or more or less depending on how many plants you're growing. And all this information is available on the Arizona Industrial Hemp page. But I just wanted to show you, they'll take a zigzag or sawtooth pattern through the field, randomly sampling plants. One thing to keep in mind is that the USDA has released their interim rules for hemp production. So these have not taken into effect yet. We're in a comment period. Well, actually, that comment period just closed. But if it passes, this will change. That typically, and before we checked for delta-9 THC, Now they want to check for the total THC. Over 900 people have commented during that period arguing against this. Because, as we saw, if 20% nationally are already testing hot at the 0.3 Delta 9, people are worried what's going to happen when we change that to total THC. So what that is going to include is not only your Delta 9, which is your processed THC, that's your psychoactive THC, it's the decarboxylated THC, but also your THCA content. That's the precursor of it. I won't go too much into it, but they're looking at this measurement of uncertainty, and I'm happy to explain more about it. But basically, they want to know how much variability there is in your lot, and if your measurement falls within that range of variability. So if you're 0.3, like if you have 0.35 Delta 9 THC, but your measurement of uncertainty is plus or minus 0.06, well, 0.3, the legal limit, falls within that range, so you're good to go. But just something to keep an eye on is these interim rules, and as they change, and I'm sure the Arizona Department page will update as it goes. But not to be discouraged, because there's a lot of potential great to come out of this industry and the research. So another topic of future research are the health impacts and benefits. We're just starting to learn of this, and largely because no one could study it. Universities weren't able to work on it, both from the agronomic perspective, but also on the medical side. So, but already we're seeing research coming out about its anti-seizure effects, it's a neuroprotective, anti-cancer, and I think as more research goes into it, that's why you're going to see an increase in hemp sales. For example, it's projected that the US-based market will increase from 390 million to 1.2 billion in hemp-based sales, or CBD sales. So that's encouraging, and I think, let's see. Another way the industry might grow is through all the many alternative uses of hemp. And this has been really exciting for myself to learn about, of not only do we have CBD hemp-based products for health, but also it can be used in construction materials, in the making of textiles. You can eat the seeds, they're very healthy. So there are a multitude of alternative uses that can benefit growers too, because it might not just be through growing CBD hemp that seems to be dominating right now, but you might be growing for fiber or for grain. Why can we do this? Because there are different kinds of hemp. You have high CBD hemp, you have fiber hemp, and you have grain hemp. And of those, you have auto-flowering. Now, only the high CBD at the moment are the auto-flowering types, so these are not photoperiod sensitive. They have a set maturity date. And then you have photoperiod sensitive. So different kinds, different ways to grow it. With the fiber, what's interesting is with the trade deal, of the 200 commodities that China has to increase their imports of, the hemp bast fiber is one of them. So this is encouraging of that this might be a new industry in true hemp, which is the light-colored, strong fiber inside that used to be used for rope making and still is, and might be a new industry. What you do need, though, is a decorticator for this. And there are a couple of factories that have opened up. There's one in North Carolina, and there's one set to open in Texas in 2024. And some farmers, what they've done is formed cooperatives and all gone in on a processing facility. So anyway, I just bring this up in terms of that there are some unexplored opportunities here. And this was shared with me by Robert, and this is a very helpful resource because though there are all these useful and alternative means of using hemp, the price is what really matters. Is it economically viable? And so this website, Hemp Benchmarks, provides wholesale hemp prices. Right now it's for CBD, flower, and the distillate, but maybe they'll get into fiber and these alternative measures. But this is a helpful resource of knowing, is it worth growing? And lastly, I'll end on another research topic, which is the phytoremediation potential of industrial hemp. So phytoremediation is the decontamination of soil and water by plants that can absorb or break down the pollutants. So heavy metal contamination in Arizona is a major issue because we have a lot of extractive industry and also just soils that tend to be naturally high in some heavy metals, like cadmium. The projected costs of cleaning up a lot of these polluted sites is very expensive, projected $2 to $5 billion for just the abandoned mines. And we have 9 National Priority Superfund sites. So these are sites that are considered by the EPA to be the most severely threatening to the environment and human health. There's one actually right here in Yuma, which has issues of soil contamination from asbestos and organic solvents in the groundwater. So it's a huge issue. It particularly affects Native communities as well, where a lot of the mining and extractive industries have gone on. It affects everyone. So typically, expensive and chemical methods are used for this, but we could also use plants. And this is a really unexplored area of research. Usually they use these like mustard family plants, and they are really good at accumulating a lot of heavy metals, but they don't have deep root systems. They take a long time to grow often. So hemp is a plant that researchers are increasingly looking into. One thing to keep in mind though is if it is doing that, what's going on in your soil? And perhaps having a soil test. This has come out in the CBD industry of, oh, that's great, hemp is a phytoremediator, but is it also uptaking heavy metals in soils that we don't want it to? So far in some research, they found that it can take up chromium, cadmium, and it's sequestering these heavy metals in its roots, which prevents it from leaching into groundwater. They also planted cannabis around the Chernobyl site in Ukraine. So that wasn't officially reported, but they say uranium and lead was taken up. So again, more research would be great to look into this. And currently in New Mexico State, they're looking at using hemp for remediation of Superfund sites with radioactive materials, and then using that hemp for biofuel production. So that's pretty exciting. And hopefully we can get something like that going here in Arizona. So there's an urgent need, I think, for this, just given the level of this heavy metal contamination. So thank you very much for your time, and I'm happy to answer any questions.
Do you see any difference of when THC through its maturity and CBD? Do you see any difference of when CBD and THC develops through the hemp's maturity? Like does CBD and THC develop in different times, or do they both develop at the end of the cycle?
It seems I can't say 100%, but it seems they develop at similar. He's asking when do CBD and THC develop, and there's at different stages. And I can't say for sure, but from what I'm seeing, it seems that the THC seems to peak at trichome development and a certain stage of that, and then it goes down. Once the plant starts senescing, you see a drop, whereas the CBD keeps increasing towards the end. So I think that's where it's cost prohibitive, but repeated testing throughout the season is allowing people to really zero in on when is a good time. And having markers like trichome color, you need your magnifying glass, but looking at that, and often the information that's out there is for cannabis, so they're trying to maximize THC, whereas in this case we're not. So, yeah. Yeah.
Is there a correlation to detect the THC and CBD in the plants with any of the micro or macronutrients?
Is there a
correlation between the elements? Because that would be a lot less expensive to test for that rather than going through that expensive.
Right. I'm not aware of one. If you can use macronutrient testing as an indicator of THC or CBD level, I think the issue is that these are just developmental processes in the plant. And if you I don't know if a deficiency or an amount really affects that the plant is going to produce these no matter what on a given time scale. But that would be interesting to see. Yeah. If there's if there's more phosphorus, would that encourage more CBD production or something? Yep. Yep. Yeah.
Listening to your presentation was excellent.
Thank you.
But it sounds like there's still a lot of research that needs to be done.
Yeah.
And for a farmer considering this, maybe, I don't know, I'm thinking, wait and see what happens. But I was wondering if we can, is there an opportunity for us as farmers to participate in any of the research with our land?
Yes, there is. And I think that's the key to this happening, is that open communication between farmers, what worked, what didn't work. So we don't have to do it all, and you don't have to do it all. I think Bindu, I know, is looking for farmers to partner with in her best management. So for everyone out there, that's someone to talk to. Robert as well. The more work we can do on farmers' fields, the better, because then it's for real, and not always in the ag experiment stations. Yeah.
Yes,
Yes.
I have a question. I'm from Oregon, and I happen to be a hemp producer, one of the hemp producers in Oregon. We produced about 50,000 acres in Oregon this year, and all the salesmen come out and sold the idea of planting hemp, which we were one of the participants. But our problem now is that we can't get any money, and we've got all our money invested, and your projections up here show how growth is going to grow for demand.
Yeah.
But we're all sitting on our product, and we got millions of dollars invested, and the contracts we have, the guys don't want to live up to them. So if there's any buyers in here who want to buy some hemp from me today, I'll be here. I'll be glad to talk to you, or you can come to talk to me. I'll make a deal with you. So, what's your projection here? Are the farmers down here, are they getting paid?
That I'm not sure, and I think this happened a lot to people, and what's unfortunate is at least you had a contract. A lot of growers I know didn't have a contract. They saw the price per acre, they grew it, and now they're sitting on barnfuls of hemp.
Well, that's what we are. We had our contracts, but like my boys say, we'll sue them if they don't pay us. And we are one of the few, most of them planted on the open market, and the paper's no better than it's written on. So how are you going to sue if there's no money out there?
Yeah.
So what I'm wondering is that are we overproducing this stuff? I just had a phone call this morning saying we have 3 to 5 years supply of hemp in production that's out there that we were sitting on. If the usage no more produced, the usage is the way it has been, it's going to take 3 to 5 years to eat up our production right now. So this projection you show up here about China and everybody's going to buy, is that true?
Well, that's for the fiber,
I mean,
right?
I'd love to find a buyer today if there's one in here. I got 3 buddies beside me here. I got to leave in about an hour and fly back to Oregon. So 3 buddies here.
This man needs a buyer.
I'll be glad to talk to you.
Yeah. What's your name, sir?
We have someone from Extreme, Steve.
All right, your name. I'm going to say it on here so it goes on the recording.
My name?
Yeah.
McKee Farms.
McKee Farms is looking for a buyer of his hemp. Yeah.
No, I'm not a buyer. That's it's an auction. I just wanted to say something, but I think this is. I've been involved in this market for a couple of years now, and just wanted to address the idea that, unlike any other agricultural commodity, you don't have all the infrastructure for the supply chain, right? So we're kind of seeing a dichotomy of the market. One being the farmers with not being vertically integrated, not having your cooperative to be able to process and get that supply chain through to the buyers, which are, as you see more CPG brands come online, which will be with, obviously with the FDA opening it up, but I've seen variation in pricing. One being, so like 45 days ago, I saw $3.10 per percent per point. You're not seeing that on the open market. That's coming from research and development from food, beverage, and supplement companies. So what we don't have here is that whole supply chain, but those that are vertically integrated, or where I think the power of the farmer is in those cooperatives, having the ability to come together, making sure that process is available to produce a derivative of a higher value than biomass. Taking biomass to the open market is going to be kind of a bloodbath. And this year, I think will be, depending on what the, obviously all the states and all the new regulations and what that looks like, it's going to be, it could be pretty nasty. However, that being said, if the FDA does approve CBD as a supplement, as an ingredient, then you could see it open up a lot more. But I just would, I guess, err on the side of caution of saying, look at your entire process and look at the very back end. And if you don't have a buyer lined up, whether it's smokable flower, something with a higher margin, the CBD market is a market. It is a good market, but it's also extremely volatile. And there is stable pricing and there's unstable pricing. So it all depends on your process, your supply chain, and how you're going to get it to the market. And if you don't start at the end, you will be sitting on it. And unfortunately, that is the case for a lot of farmers right now. And one of the problems is the processors, they're not held to buy it, right? They're basically taking a split. So whatever you grow, typically you're already losing part of your yield based on the fact that they don't have to buy your biomass from you. Like they're just going to process it and they have access to the buyer. So a lot of success, I would say, from some of the smaller farmers is going to either a finished product somehow, processing it even on a small scale, but just understanding that you have to start at the very end of this whole thing, and or else you will end up sitting on biomass like this. But there are buyers out there, and there is a good market. It depends on where you go, but it's not one of those things that you just jump into just to grow it without having that plan available to you. But anyway, sorry, I'm with that, Adina. We're one of the nursery programs here. I'm happy to talk as we go through this process today.
All right, so our next speaker we have is coming all the way from Imperial County, where he works in the extension department there. Dr. Ollie Bocci, who, did I say that right?
That's right.
All right. He's here to talk to us about pollination, cross-pollination and pollination of your hemp, and things to watch out for.
While we are just under plan to study the extent of pollen drift in industrial hemp, Dr. Bindu asked me if I could speak on pollination and the cross-pollination issues, and if those are friends or enemies. I am very happy to be here to talk or give you information or just a reiteration of information that you already know on pollination and cross-pollination issues. So, just some clarification that cannabis usually describes the cannabis sativa, which is bred for high THC and known for its psychoactive material. On the other hand, industrial hemp is described, it's the same plant species that contains only trace amounts of THC, and it is used for industrial purposes. And those are products, three main products, even though it is versatile, and they say they can extract more than 250 products, and so they can be classified into, those. The fiber, oil, or oil seed oil, the food and feed, and then, of course, the most desired CBD. All right, so the pollination dilemma is that, number one, we need to understand that those plants can be dioecious, meaning they can have separate male and female plants, or they can be monoecious, meaning they can have the same male and the female flowers on the same plant, right? So male, hemp plants produce pollen, which fertilizes female plants in many ways of pollen transfer. So you can see in here, okay, it could be a selfing or self-pollination, which means the same plant because it has, male and female. It could be pollen from the same plant, but on different flower. On the other hand, pollen can transfer from one plant, separate plant, to another plant. So those are three ways that pollination and fertilization could actually occur. So a single male flower is known to produce about 350,000 pollen grains and pollinate female flowers. So that means even if you have a single male plant, it can immensely affect the whole field. So it's a lot to look at. So pollinated flowers produce seeds, and then the plant starts to invest energy in seed production, and hence less CBD. Why? So that takes us, this is actually the male flower in here, and then releasing, the pollen in here. That's what it looks like. So studies actually suggest fertilized females can actually produce 50% less CBD than or relative to the female that is not fertilized or pollinated. So it's a big loss by being pollinated. So CBD is primarily stored where? Of course, in the trichomes of the female cannabis flower. All right. So what are the reasons that a plant would produce less CBD when it is pollinated? Okay. Now, look back to our knowledge of crop or plant physiology. And when we look at that, plants can be generally divided into a source and sink organs. So the source organs are the leaves that manufacture or photosynthesize assimilates. And then the sink organs, which can also be a sink and a source at the same time, as long as it has chlorophyll in it. So buds can be one of those examples. And then the seeds are actually, the sink. So photosynthetically active source organs, example, the leaves, are usually defined as net exporters, while the sink organs are referred to as net importers of assimilates. So that helps us understand now why would a fertilized female flower that develops seed would actually lose CBD production. All right, so pollination and seed formation. Now, in crop physiology, we know that seeds are a strong sink. They act or affect assimilate partitioning to storage organs. Now, the buds where the CBD are stored or the seeds of the plant. So with the strong sink then, the seed, CBD production, which is concentrated in a female flower bud, of course, it suffers resource allocation and the yield reduction. So this is simply a competitive analysis between the two, zinc organs. Okay? And that seed is stronger competitive than, the bud. That makes it then, for the plant to be of less CBD producing. It concentrates into partitioning the assimilates into the seed rather. So many researchers stated zinc strength and the competition for assimilate attraction that determines yielding potential of that organ. And some said in the reproductive structures, the zinc or seed becomes more active and shifts to a higher potential when only the plant is pollinated and fertilized, or upon the formation of the seed that becomes strong zinc. Therefore, we know that male plants are liabilities to CBD farmers in that pollination and heavy seeding dramatically reduces CBD production. And hence growers must be vigilant to check their field for male plants and take action. Believe me, I'm not cursing male flowers or male plants because they have their own values. This is not the topic of the talk, but of course we know that the male plants would have, or are stronger for fiber production, seed production required, and then biodiversities or diversities and so on. So that being, pollination issues, how about cross-pollination? Remember we said another plant with another plant. Now, while this would actually produce the same effect as pollination, it doesn't matter whether it is cross or self-pollination, on CBD production, cross-pollination would actually have additional liabilities. And those are in two ways. One, if you have got an industrial hemp plant that produces pollen, and there is a nearby marijuana or the ganja, right? It destroys that marijuana commodity. Why? Marijuanas are produced for or optimized for high THC, right? The high psychoactive material. And if a low THC male fertilizes the desired high THC producing plant, THC goes down, the market goes down, okay, for marijuana, meaning there is no potency and the value of selling, medicinal or adult use products declines. And also have another, the reverse. If you have got a male marijuana plant, of course, industrial hemp farmers would not like it, right? High THC causes of a marijuana, male causes or rises the THC level, which makes THC levels of industrial hemp above the 0.3, limit. What happens? The whole field should be destroyed in order to comply with the regulations. So that is what we look at. One, CBD production. Two, effects of increasing or decreasing THC levels in plants. All right, so there are studies to look at the pollen drift. This is actually what we look at using the model of spore release from microorganisms, which would be the same as, the pollen. And we plan to work at what is the zoning, what is the distance or buffer zone between plants. It's a dilemma. Nobody knows exactly the extent because it would be variable depending on environmental condition, the strength of the wind, or the activity of pollinating agents, bees and others, right? So anyway, the studies have shown, some studies showing pollen drift across 3 to 7.5 miles, meaning, hey, you can actually grow, or it is a kind of, you can't be my neighbor if you are going to move in within this distance. Okay? And of course, it has happened, and there has been litigations, the studies or the information in Oregon, a multi-million litigation against each other. So another study actually showed it's 30 miles. So look at this. This is a big variation between the two distances. So we are not really very sure as to if there is an ordination to enforce some kind of zoning. No clear understanding. All right. So with having said those, what are the methods that are used or recommended to control or suppress pollination and cross-pollination issues? Number one, in general, there are not that many outdoor marijuana production, right? So that is a good thing. Okay? That marijuanas are mostly indoor and therefore may not have an effect with or interference with the industrial hemp. But that still depends how tight is that indoor infrastructure. Pollens are very tiny. They can actually pass through the screens, right? So the other one is growing indoors with air filtration system. In fact, this is not actually a complete control because of the size of the pollens that can escape, through the screens or the filtration system. In some other areas, people just surround windbreaks, okay, with other tall plants. One of the good examples is sunflower. They can plant around, their, industrial hemp fields. Okay? And this, again, is simply a means by which to suppress the possibility, but not an absolute control. All right, zoning establishment. This looks, working. It has worked in, in Colorado, a good example in developing, zonings. So they have got female hemp, male, or hermaphrodite, hemp. Those are those that have both, male and female organs. So the examples are, the establishment of industrial hemp in only certain areas. So who would do this? And of course, zoning would require the ordinance from local, state, and federal government in association with the growers. Okay. All right, so this is too bad. Deadhead, the male, plants. And that is, of course, a problem as well. How do we differentiate male marijuanas or male industrial hemp from the female? One of the good things is the males produce pollen or flower in 7 to 10 days before the female flowers. So we have got time to, cull, the male plants if it is desired. So how do we know male plants? They say in most strains, male plants have thicker, stalks than the female. And they produce relatively larger leaves than the female, which has smaller leaves, but many of them than the male plant. And another one is to look at, the buds that grow between nodes. Now, the male, as you can see in here, has no hairs on the buds, and the female do. So that makes it, a little easy to identify the male from the female. But is that true for all strains of industrial hemp or marijuana plants? I don't know. Okay? But this is, what has been said, just to help identify the male and the female before you destroy the good plant. All right? Okay, so staggered plants, meaning you can actually grow non-friendly commodities at different times. Okay? Not only different locations. This can be looked at analogically by looking at the differences between field and the sweet corn. They can interbreed, right? But for different purposes. So now the field corn has a dominant gene as opposed to the recessive gene types in the sweet corn. So if that dominant gene pollinates the recessive gene, what happens? The dominant would actually show phenotypically, right? All right, so what do people do? They don't usually grow field crop and the sweet corn in the same proximity. Or they actually plant them at different times. They plant the sweet corn during, spring or winter, and then the field crops late in the spring. That's what, they do in Imperial. Okay, so another one, growing sensamilas, the Spanish word, meaning feminized. I'm sure you know all these things. Okay, and those are, desirable, of course. Yes. Now, strictly male plants, that actually means that the plant produces the desired CBD, and it doesn't have to waste energy to take care of the seeds. Okay? So that is, that makes it good. But of course, there are shortcomings. You can't be happy all the way. Why? The amount of money that you pay for feminized seeds is by far higher than the ordinary or the conventional ones. And the number two, most of them can be snake oil. You buy for having a female plant, and you may discover a lot of male plants when you did not expect. Did it happen? Yes, it has happened in many. And know that even if there is one male in it, produces how many pollens? 350,000. It's good for the whole female in the field. All right? Okay. So, also suggested are misting male flowers early morning. What is this doing? It simply makes the pollen heavier and not to be able to drift further. So people are doing it, But would this be possible in a large-scale farming? You get up in the morning and then, of course, rinse the plants, the whole field. That's maybe not practical. And another one is spraying anti-ethylene compounds. What is ethylene? Ethylene is naturally present, and it enhances flower production. So by applying a chemical that suppresses it, you're making the male plant not to be able to produce as much flowers as possible and contaminating large mass. So with this then, in summary, self-pollination or cross-pollination causes fertilization and the seed development that becomes strong, assimilate, and outcompete other storage organs. Poor resource partitioning and then energy allocation to flower buds. And then the CBD storing organ results in lower CBD production. Okay? If it is, poor partitioning as a result of the loss of strength in comparison to the strong seed next to it. All right, and therefore, male plants are liabilities or a hindrance to CBD production, and then the plant's THC levels. Vice versa. We discussed, this. So, avoiding pollination or crop pollination could somehow guarantee that female plants produce desired CBD amounts, and the THC level in all cases stay reasonably constant, meaning it is good desired level for the marijuana and then for that of the industrial hemp. So, of course, there are a lot of uncertainties. Even though industrial hemp is not a new crop, well, it has grown since 1920 in Imperial Valley. But since, the banning, there is not, information, anything on breeding, on productivity, and so on, even varieties. And then, of course, how the industrial hemp would actually behave under the dry, hot conditions of the low desert. We have no information if a dry condition, high heat, would actually modify the THC content of a plant that has got a complying THC level in the temperate zone. We don't know. This is something that we need to study. We have got a lot, jubilations to work on many, vacant research areas of industrial hemp. All right, so the research is necessary to quantify the extent of pollination or pollen drift, variety selection for desirable CBD, productivity. And of course, I believe that future breeding techniques may enhance either the source strengths so that it can partition to the competing organs, zinc organs, or improve competitive strengths of the CBD storing organs. Okay? Those are something that, we just, forecast could happen. Okay? All right, so with that, this is, my hideouts. This is the Desert Research Extension Center, and that is the Office of the Cooperative Extension, and all this area is the 255 research acre of the University of California Agriculture and Natural Resources. we're planning to bring research in the future of industrial hemp into this. Okay, so I think that is all I have. I thank you so much for your attention. I would be happy to answer if there are questions and I know how to answer them. If I know, yeah. All right. Yes.
Holding, can you speak to your plans for your future research in Imperial? Are you going to run concurrent research with what the U of A is doing, or are you going to have a different focus?
Well, actually, we have started to work on industrial hemp research as of 2017, long time ago. And then here we go. There are, regulations at different federal, state, the county, and then the university itself. When everything became easy, it was not easy with our university to give us a green light to do research. So now I think we are very close. And then, of course, we would be able to do research on the university property, but not any kind of a support on a grower's field. We are awaiting, after actually writing memorandum of understanding with the growers, and then backing off so that we cannot actually work. So there are many that we have planned to work on. Simple variety trials, and then of course determining water use or water use efficiency of industrial hemp under the low desert. And then of course variety trials using high and low density variations. And one of the things that we actually plan is the growers are interested at the availability or the condition of the low desert that have got 365 growing days. But we want to know, by having different growing seasons or planting dates, and see if the plants can actually be, or multiple cropping and harvests can be done under the low desert condition. I think that should give us a competitive advantage than those areas that do have a single growing season only. All right? So when the time comes, hopefully, and the money available, we are planning to do all those things. I think we will collaborate with people from here. We have been in connection with, contact with people. So we will work, on those. Those are the areas that we plan to work and research. Any other? Yes.
Do you know much about the CBG components in the hemp compared to the D? Like THC is for marijuana, THD is for pain, this type of thing, or relaxation. D or CBG. Do you know anything about that component?
No, I know there are various products, and then of course the bioproducts after which actually develops into, but I'm not sure as to any of the chemistry of those products. And maybe yeah. Okay.
So I'm not a chemist, but I can tell you just a little bit. But basically, the gist of it is CBD or CBG is the precursor molecular compound to multiple cannabinoids. So they call it the mother of all cannabinoids, and that at some point through the pathway, and biochemists, please don't correct me or embarrass me, but eventually it can and will convert to THCA, THC, CBDA, CBD, and that's about all I know. But it's a precursor molecule. There's some evidence potentially that it's a antibacterial potentially. It's also, I think, interesting to neuroscience. And that's about as far as I'm willing to go with it. But that's my understanding, and so there's some, it could be potentially multifaceted.
I have a question for you. I've heard the CBDA can evaporate. Is that true?
I don't want to, I don't know. I couldn't tell you. It's about the extent of my, there's just not a ton of, I'd say as much study or research as there available as there is with CBD and some THC. So CBG is relatively new. I think there's a lot of excitement around it. I will say from a market perspective, it does have a much higher market price right now per percent per point. The hemp benchmarks, I think if you got a chance to take a glimpse at that, CBD right now is anywhere between 50 cents per percent per point up to, say, $1.50. On the open market, again, that's open market pricing. CBG is currently around, ranging around $15 per percent per point. So that being said, it does have a higher value on the open market currently, but at the same time, who's your buyer and what's the use for it?
For growers, could you tell us how they calculate, like, the percent per point per kilo, or how does that work?
As far as how do you get to the derivative?
How do you get to that price? How does that calculate to get that price? I mean, I'm going to do it. I want to know how I get that price.
Yeah.
That's my question, and I'm putting all the questions, but I need to know how to get that.
It's really, so it's, I guess, it's a supply and demand market. So with CBD, you know, again, open market, we saw, I think, an oversupply, and this gentleman from Oregon, you know, mentioned that a lot of people are having to sit on it. And there's a whole lot of variables as to why that is, I would say. And some of it, I think primarily it is, you can say that it is a result of just the instability in the supply chain and from infrastructure perspective. So basically if you're COA, you're testing your crop, and then you're testing once you get your COA, your certificate of analysis, you'll see what your cannabinoids are measuring. So you'll see THC, hopefully that's well under 0.3%, CBDA, CBD, and so you'll see what your total cannabinoid makeup is. And then based on those, what CBG is showing, if it's at 5%, and then the market price is speculatively at $15 per percent, then per pound. So if you produce 5% CBG and you have, you know, a pound of flower or biomass that comes from that plant, then you know, 15 times 5 is essentially your price per pound, theoretically. Again, it's still also is only as good as, I guess, what your buyer will give you for it.
Is that pound whole plant or just flower?
So typically you'll do a whole plant to some degree. There's various, I guess, makeups of selling it. So either you can do just flower, aerials only, colas only, whole plant biomass, including stalks, stems, everything.
The reason I said all these farmers here, they can go and take a core sample of the hay bale at the bottom of the hay bale and get a couple of protein out of the hay bale and sell it for premium, or they can take a core sample out of the middle of the hay bale and get the real reading on it. And I mean, we as farmers could screw you guys over so fast. I mean, I know I could, but based on COAs.
Right.
But we need to know as a farmer what's going to be the truth so that we can actually have an idea.
Right, and that's, you brought up a great point. I think that's a huge issue just in the industry alone, is that no matter if you're buying anywhere in the supply chain, if you test these plants or somebody's seed or whatever, the COA could be false. I mean, and you see a lot of that out in the market. So I mean, I think very extensive due diligence throughout any area of the supply chain. Yeah, you're absolutely right. And a lot of people do get screwed. I mean, that's the unfortunate fact at the moment. And so that's just where we are until we get that true stability that we have in other agricultural commodities. It's really know who you're working with, do your due diligence, and protect yourself as best as possible.
Also, we have somebody here from Northview Extractions. Would you raise your hand?
Steve?
Steve, would you care to comment on that? So he's a purchaser for processing.
We have a new extraction facility coming up in Phoenix, so we're able to, process the biomass.
So are you buying biomass?
Not at the moment.
So our next speaker is coming from Tucson, where he's Department of Biosystem Engineering. Dr. Cuello is going to speak to us about hemp cell culture and sustainable alternatives to open field pollination.
Thank you, Robert. Thanks to Bindu and Robert for getting us all together here. Thank you all for showing up here today. So my presentation is really by way of information to introduce to you an alternative method of producing the biological products from hemp, meaning to say the cannabinoids and perhaps even the terpenes, which are starting to be recognized for some of their beneficial pharmacological effects as well. So I'm going to talk about particularly cell cultures. So I'm sure we all know why we're here, and most of you are familiar with this. Hemp, industrial hemp, is well known for its promise for green wellness. And the CBD market by 2021 has been estimated by experts in the industry as amounting to about $2 billion in terms of revenues. And Dr. Carter gave a much more conservative figure earlier this morning. But this is one particular expert in the industry who's more generous in his projection of the size of the CBD market by 2024. And he's pegging it at about $24 billion, which is really quite sizable. So why CBD? And I'm sure most of you are already familiar with the promise of CBD. And these are some of the claimed benefits of CBD, which still are under research. You've got addiction, aging, anxiety, all the way to PTSD. So it's a very promising compound. And it's not just CBD, because the Industrial Hemp produces over 113 different types of cannabinoids, THC, CBD being two of them. And the others are not very familiar to the public yet. And a lot of them still have unknown potential benefits for pharmaceutical or pharmacological use. One thing about open field cultivation of industrial hemp, particularly if you're focused on producing the cannabinoids, is that there's a lot of variability. And these are the major factors that affect the mix of cannabinoids as well as the relative concentrations of those cannabinoids produced by the particular plant that you're growing. So that depends on the specific variety, and of course the individual plants as well. You've got the sex, the age, developmental stage, climate and weather, cultivation methods, time of harvest, and storage conditions. So when you're growing the hemp plants outdoors, it's subject to this variability of all of these factors. And so, again, you cannot guarantee the specific mix as well as the relative concentrations of the cannabinoids that the plants are producing. And that is why the earlier speaker mentioned that a big percentage, I think about 40% of the industrial hemp grown in Arizona recently has a THC concentration of over 0.3%, which is the ceiling. And so that could become a problem. And that's one of the disadvantages of growing in open field cultivation. So an alternative, of course, is to grow indoors. And I know there's at least one company here that's growing industrial hemp indoors, not in a greenhouse, but in a warehouse type of facility. But there are other alternatives to that, and that's what I'm going to focus on, and using the techniques in biotechnology, and that is using cell cultures. But before that, one technique that's now being used in industrial hemp from biotechnology is plant tissue culture or micropropagation. And I believe the next speaker is going to talk about this. So this is only by way of introduction. So micropropagation really is just about cloning. So for instance, if you're working with a particular variety and you have a particular individual plant that you really like in terms of its vigor, in terms of its growth rate, perhaps in terms of its cannabinoid profile, you can actually clone that particular individual plant, and that is using plant cell culture. And this is a general method of accomplishing that. So you've got your potted plant, and you isolate a particular part of it, and then you go through these aseptic processes. I'm not going to go through this, but that's how you actually produce a plant in vitro, meaning to say in a closed environment, in glass. And it's aseptic, it's sterile condition, so there's no contamination, and you manipulate the hormonal levels so that you can actually quickly micro-propagate or multiply the number of the plants over a short period of time. And those are genetically identical. So even in open field cultivation, micropropagation is actually recommended long-term so that you can maintain the particular individual plant within a given variety that you really like in terms of its features. So you can be sure that you're not going to lose them. So these are not hemp plants. These are plants in the horticulture industry because this method is now widely used in the horticulture industry. So this is one thing very interesting about plants is that it's very easy to manipulate them just by manipulating the hormones that are present in the growing medium. So these are the two major or basic hormones for plants. One is cytokinin, the other one is auxin. And by manipulating their relative ratio, you can actually direct the plant to assume a certain form. So if you have a cytokinin to auxin ratio of greater than 1, in general, you can produce only shoots without roots inside that controlled environment vessel. If you have cytokinin to auxin less than 1.0, you will be producing only roots without the upper parts of the plants. And in general, if they are equal, cytokinin and auxin, you're going to produce undifferentiated cells. So this is my major interest here, is to produce these cells and then to propagate the cells in a bioreactor, which is a controlled environment vessel. Again, these are pictures of horticultural plants that are being grown in vitro for micropropagation purposes. Okay, so the next technique is root cultures, and I actually worked on this. Well, for my master's at Penn State, I worked with micropropagation, and then for my PhD, I worked with hairy root cultures. So there's one group of researchers here in the US that has worked with expressing CBD and THC in roots. So you're not growing the entire hemp plant, you're only growing the roots. However, the roots have been genetically modified with some bacterium and introduced with certain bacterial genes, and hopefully it would express the CBD or the THC as well. They succeeded in, and this is not roots of hemp, these are other roots, but just to give you an idea what it looks like. So again, you're not growing the entire plant, you're just growing the roots, and you can grow them in a liquid medium, and you can scale it up by growing them in bioreactors or scalable vessels. And the hope is that it will produce the CBD and the THC. So that particular group that worked on this research, they were able to produce CBD and THC, but only in small quantities. So obviously, further research needs to be done in this area. All right, so now let's focus on cell cultures. Again, the way to produce cells out of any part of a plant, whether from the leaves or the stem or the roots, is just manipulate the cytokinin to auxin ratio. These are plant hormones that you regulate in the growing medium. And typically when the ratio is 1, that's when you produce the undifferentiated cells. So of course, this is the inflorescence picture of a cannabis plant. And once you have isolated the cells, you can grow them like a normal plant. So it requires CO2 and air, it will require nutrients, it will require water, and depending on what plant or plant tissue or material you derive the cells from, it may need to continue photosynthesizing. So if it is photosynthesizing, it's green, you need to provide the light. All right. So what are the advantages of growing cells instead of the whole plants? So there's a lot of advantages actually. One is you don't need arable land. You don't need a farm. You can have water savings, significant water savings. And the approximate valuation here for water savings is about 80% compared with open field cultivation. Nutrient savings, because you can keep reusing your nutrients. Your nutrients are not going to be leaching through the soil and potentially pollute the environment as well. It becomes weather independent because you can grow these in a warehouse type of environment, in a laboratory or a warehouse. No pesticides. You can really regulate the input chemicals in your system. So no herbicides, no pesticides, no assimilation of toxic substances from the soil. Dr. Carter mentioned that industrial hemp is an excellent phytoremediator. So if you happen to plant your plants, the hemp, in a soil that is polluted with heavy metals or some other toxic substances, they could uptake them. And they could be accumulated in the stems, in the leaves, and other parts of the plants. So with cell cultures, you don't have that risk or potential danger. Definitely clean and safe production because it's a closed system. And here's something very important for business is consistent and reliable production. So once you're able to figure out the set of environmental factors where you're able to optimize your CBD and your THC is less than 0.3% or 0.3, then you can stick to that set of environmental factors and you can be sure that your cell cultures consistently will be operating within that range of those chemicals of interest. And scalable, which is very important, meaning to say you can scale up, you can enlarge your operation to whatever scale you want it to. So here's a representation of what this would look like. So full disclosure, we're just about to start this research, and it's going to be sponsored by a company based in Phoenix called Taurus Ag. And I think I failed to acknowledge my co-author, Yasser Medipur. He is a research scientist who'll be working on this project in my lab. So the idea is, once you have successfully isolated and initiated your cell cultures from the hemp plant, you can grow them in flasks in the lab. But it's hard to scale this up using flasks to produce economic volume. So you have to grow them in bioreactors such as this one. And of course, the bioreactors can be, you can have hundreds and thousands of them in a facility, which could be a greenhouse or it could be a warehouse type of facility. So it becomes like an industrial factory for producing the biochemicals produced by industrial hemp. That's the model. All right, so here's a greenhouse at the University of Arizona, and these are some of the bioreactors that we developed. These are original bioreactors that we designed and developed in my lab group at the U of A, and they're actually patented by the University of Arizona. We call this particular one the air accordion bioreactor. And again, this is not CBD cells. These are not hemp cells. These are algae cells, but the operation will be very similar, if not exactly the same. So this is a time profile. This is day 0, day 5, day 7, day 10. And you can see that the intensity of the greenness is increasing because the cell density is actually growing over time. So this is what it would look like. And this is just showing you the growth curve of the cells over time. And of course, we want to be able to stick to this period here because this is where the maximum growth rate is increasing. And there are techniques by which you can maintain that. For instance, you can harvest part of the cells once they have reached the maximum growth rate and they start to saturate, and then repeat the process so that you're able to maintain your maximum growth rate and productivity over time. So just to show you some of the bioreactors that we have designed that would be appropriate for this application, one is the regular accordion photobioreactor. This one is made of polyethylene bags, and there's a pump here, and so it circulates the fluid up here and then down here, and then back into the plastic bag. This one is the air accordion, so instead of the liquid recirculating, the liquid stays inside the plastic bag, and then there's an air sparger at the bottom, and you bubble air through it. And we introduce CO2 there as well because that helps the photosynthetic process of the plants, the plant cells. So you can manipulate not just the aeration, but you can manipulate also the nutrient concentration and mix within the bioreactor. And these are some of the advantages. Low power demand, low material cost, ease of scale up, and so on and so forth. And depending again on the cells that you're using, it could be photosynthesizing or it could be non-photosynthesizing. So here's an example of cells that are photosynthesizing. They're green, they need light. Or these are examples of cells that are non-photosynthesizing. They can grow in the dark in the absence of light. Here's another type of bioreactor and another type of bioreactor. So we have a lot of bioreactors that we can use for growing these cells and mass producing them. So again, it matters where the cells are derived from. So we all know now that the THC, CBD, all the other cannabinoids, they are produced in the trichomes. So, of course, if we want to start cell cultures, we want to derive our cells from this part of the plant. But as you know, there are varieties that accumulate CBD and THC as well from the leaves. So that is worth investigating as well. And if it's from the leaves, it has to be photosynthesizing, so we need to provide lighting so that the cells would grow appropriately. So again, I just want to repeat here the advantages of producing the biochemicals from hemp using hemp cell cultures instead of growing the entire plant. And I would be happy to answer questions. Thank you.
This is a basic question. I don't understand how tissue culture works, but so say you took the trichome cells and you were able to grow trichomes.
Yeah.
They don't have any plant or leaves or roots, it's just the trichome?
Right, it's just going to be the cells of the trichomes. And actually the trichome is a tissue. It's composed of different types of cells. You can actually focus on specific cells within the trichomes as well.
So if you use that bioreactor, you would take some of those cells out and then grow them separately?
Yes.
So you can't grow in that water.
Right, so you take the cells and you grow the cells in a liquid medium with a proper nutrient mix. And the cells will multiply. They will divide by mitosis. So it's asexual multiplication. And so you can grow them in flasks. But of course, for commercial operation, you want a larger volume. And it's more efficient to grow them in what's known as bioreactors or photobioreactors. So just so you know, different research groups have already successfully produced cell cultures from different tissues of the hemp plant. So that's been done. That's not a challenge. The challenge is making sure that those cell cultures actually produce the CBD and the other cannabinoids. And so our focus is making sure that they do, those cells, and that they optimize them. And we have different techniques as well in terms of maximizing and optimizing the production of these chemicals in the cell cultures. CBD and the other cannabinoids, they're referred to as secondary metabolites as opposed to the primary metabolites. Primary metabolites are proteins, carbohydrates, lipids, whereas secondary metabolites are special chemicals. They're fairly complex chemically, and most plants produce them as a defense mechanism against invading microorganisms or against environmental stress. So I'm not surprised that in Arizona, for instance, there's an uptick in the production of THC in the industrial hemp because the environment in Arizona is very stressful because of the heat. And so that causes the plant to be stressed, and as a reaction to that, they overproduce cannabinoids, including THC. So there are techniques that you can do in the lab to simulate that stress. And it doesn't have to be heat. It can be salinity, it can be ultraviolet light, it could be temperature. So when they're exposed to these stressful environmental conditions, they feel, the plants, they don't feel, but they're coaxed, they're induced to express genes that would result in the expression of these secondary metabolites. All right, questions? So just to be clear, this is not ready for prime time yet. It requires research. But this is more than likely one of the futures of biochemical production from industrial hemp because it's an efficient method of mass producing them and also a sustainable way of mass producing them because it saves on water, nutrients, and other resources. Robert.
What other crops are you looking at doing that with?
We've worked over the years. I've been at the University of Arizona now for 25 years. So we've worked with different types of plants. We've worked with agave. We've worked with tobacco as a research plant. We've worked with carrots, cell cultures, and a whole lot of variety of plants. And of course, algae cells, different types of algae cells.
So for our next speaker, we're going to have someone from Adavina Nurseries, which just so happens to be one of our sponsors for this event today. Dr. Chris Pagliarillo. Are you here?
I'm right here.
Perfect. Okay, so he's going to talk to us today about a couple different lectures, talking about hemp genetics, pros and cons of seed, clones, and tissue culture. And then he's going to roll into another one, successful hemp propagation, managing a rooted and unrooted cutting and transplant program.
Thanks for inviting us out here. We're very grateful for the opportunity, and hopefully y'all have had a chance to meet our ladies here. These are our cherry wine, Mystic cherry wine mothers that we use. So I'm Chris. Unfortunately, Hope couldn't come out and make it today. She got called to the Capitol. She's on the Arizona Industrial Program Committee, the Rules Committee, and the Science Committee. She helped write the rules for the hemp. Right now, the Arizona Dispensary Association. So she got called out because the ADA wants to establish some mandatory buffer zones around medical marijuana cannabis grows because any hemp or any pollen that gets into their grows could have a devastating effect. Now, there are ways they could be responsible about it without telling you what to do in your backyards, because this is Arizona. So Hope is going to the Capitol to ensure that whatever rules come down are in everybody's best interest. So I'll try to represent her talk as best I can. We're both products out of the Controlled Environment Ag Center at University of Arizona. We were the first generation out of there. She was my student worker. She was the only one that would help me move my 300-pound reservoirs. I figured I better marry that woman. Women on tractors, that's what I like. That's what I'm into. She wanted to tell you about tissue culture, and it's how you think about that when it comes to making decisions about if you're going to want to get into high-value cannabinoid crop. You're going to hear tissue culture. What does that mean? Why is that something you want to think about or engage in? I'm going to tell you about once you've kind of decided you want to get in this business, there are, seeds are an option, as are young plants or transplants. Why would you go one way or the other? There's not one better than the other. It just depends on what's your goal. So I'm going to cover some of the ideas there. Try to make this as useful as possible. If you have any questions along the way, just hit me up. So again, we've been in cannabis for about 4 years. 2 PhDs amongst legions, armies of bros. Kind of gets hard sometimes. Just because you spent 20 years in a basement getting high and talking to your plants doesn't mean you know plants. So it's been a little frustrating. So we're kind of excited to work with actual farmers that want to know and learn about plants. We've seen things get really ugly. We've seen all the best and the worst. We've seen entire grows get decimated by a virus in less than a few months. So we're very conscious. It's the same plant essentially, so the same thing could happen in your fields. We're trying hard, working hard to make sure that doesn't happen. Give you young plants or starters that are going to give you an edge, not screw you in the end. Adavina means kind of to foretell the future, or a truth teller. So we thought that was a good word to describe what we're trying to do. As we've talked about many times here, the Arizona hemp crop was destroyed. Anybody notice something wrong with this picture? That's not hemp. Thank you, K-Gun. Fake news. But yeah, it's a problem. We're struggling here probably because of different environmental factors that we need to take into account. What we're working towards is you can never eliminate risk, but you can mitigate it. So what are some of the things that you should think about in making the decision to get into this business? These are the issues that we're trying to help you avoid. Over and over, we've talked about today variability within a crop. With seed crop especially, I've been out in hemp fields where some plants are this tall, some plants are this tall, some haven't even gone to flower yet. The variability among seeds is insane right now. It's a very new crop. It's going to take several years before stable seeds are going to be available. Here's one of those examples of a mosaic or a virus that spread throughout a massive cannabis grow very rapidly. How do you think about that and avoid that? We don't talk about it much, but there are seed pathogen problems. These are seeds that are exuding fungal or bacterial. Their virus can live in seed coats, so that's something you need to think about. Bacteria and fungus can live in the plant themselves, the systemic problems. There's nutritional issues and blight, whatnot, all of these we're trying to take into account so that when you get a plant from us, these are the last things that you need to worry about. And once you put that seed in the ground, it's game over. There's no more preventative action that you can do. So the model goes back to the Yoder brothers. What we're trying to do is build out a stock of elite, a population of elite plants that are a reservoir of clean germplasm that you can then constantly rely on to always have a clean and healthy crop. Back in the 40s and 50s, the chrysanthemum industry was about to collapse. Everything was infected with either viroid or fungi. And the Yoder brothers reached out to Cornell, one of the first to use tissue culture, basic tissue culture technology, to develop those elite plants that have been indexed, make sure they're virus-free, pathogen-free. They jumped on board, systematized that, and now it's a basic standard in all horticultural crops. Every strawberry that you have eaten at the grocery store has come through an elite stock program that was originally based on this early work by the Yoder brothers, who eventually took over the chrysanthemum market and became the mainstay. So the issues that they were having back then are the same things that we saw over the last 3 or 4 years in cannabis. We saw massive disease, virus, die-offs, notorious, powdery mildew is very common, and we think systemic, russet mites, all of these things were becoming to take over grows and really hurt yield, and there was no game plan on how to deal with it. These viruses and pathogens were devastating the yields, and there was no way to bounce back because there was no easy and quick way to replace your germplasm. So that's what we've been, and so there was, we're hoping that this doesn't contribute to the current failures. So we're turning to tissue culture to begin to build out those elite stock, clean them up, and have them ready for you when you need new starters. As Dr. Koala quickly helped us understand, tissue culture just means you're taking a few cells or even a little piece of tissue. Plants, because of their nature, they're easy to encourage those cells to de-differentiate and redifferentiate. It's kind of like if you could take a piece of your finger and make a whole new you. It doesn't work that way in animals, but in plants, that's something easy you can do. Now, why would you want to do that? The nice thing is you can get at these pathogens. Unlike us, plants don't have immune systems that can target and eliminate bad stuff within them. Once a virus is in there, it's in there until that organism, that plant is dead. But they can with a little help from us. We can use tissue culture techniques to isolate tissue that has yet to be infected in a plant and take that and that alone grow up a whole new stock. This is a picture of kind of the general framework of a clean stock program in any horticultural crop, like be it a apple tree, pear tree, any sort of citrus, strawberry, raspberry. This is kind of the basics. You start with a great hot new plant. You're going to quarantine for a bit. You're going to look for pathogens and viruses, meaning you're going to index them. You're going to see if you can detect what's in there. These days it's all molecular, but back in the day they just used kind of like Confederate or sucker plants that they knew if one was infected, this guy would definitely get infected. They then used tissue culture to eliminate or clean up any of those that were detected. Then they make sure the tissue culture process itself didn't screw anything up. If everything checks out, they harden those off, get them ready for the real world, and now you got your growing stock of clean plants you can go back to every time you need to start over. So in cannabis, we're just getting started on this. We're just starting with where you can start. One, everything we do goes through quarantine. We have a lab in Phoenix where we can monitor and see if what's in there, if there's anything. We can pathogen and virus index, see what's exactly in there. And then Hope is working on the tissue culture protocols that will allow us to eliminate any endemic virus. So tissue culture, real quick, a little biology is possible because, again, plants have this totipotency capability. They can take one tissue and with the encouragement of certain combinations of hormones, they can make a whole different kinds of cells. These are called adventitious roots. That just means you got a foliar part or a shoot part of the plant that can be stimulated to make roots. We can take that characteristic of plants. We can start with a very tiny piece of plant, sterilize the crap out of it until it's half dead, and then give it all the media in a sterile environment once and the right hormones, and bam, we got a whole new plant that's at least pathogen-free. Anything that was inside pathogen-wise, we've eliminated. Woo! But so plant tissue culture is not new. This is widespread across the country, done like in all the crops that we were talking about. You could look online and find all sorts of, you want to start an almond orchard, you're going to go to a micropropagation company and they're going to sell you a bunch of tiny little almond starts from tissue culture. So why do we care about this in cannabis? Because for decades, all you've had to do is take your mother plant, make a cutting, then you got a whole new plant. Why not just keep that going? Well, part of the problem, besides the virus pathogen load, is that a mom is a plant that, like any plant, if it endures any stress, it's going to change its gene expression just to deal with the stress. Instead of maybe spending all its energy on growing and getting big and making reproductive parts, it's going to spend some energy on dealing with salinity or dealing with the sun, making other compounds. Now, if you keep cutting from this mother, and some folks have kept mothers for like 20 years, those cuttings remember that stress training. And if you keep compounding it, no matter if it looks healthy, it remembers its programming. It doesn't matter if you're babying it, if it's living in Wonderland. It remembers, holy crap, I'm in salty soil. I got to spend all my energy moving that salt out. So over time, these programmings take a toll on a plant's yield. And what you need to do is kind of reset that programming and say, hey man, you're going to be fine. Tissue culture, because that combination of hormones tells that those cells reboot. It's like turning your computer off, turning it back on. It's a reset that allows the plant to then rededicate it to whatever the environment is actually telling it to do. The other big advantage with, instead of having moms, is that these take a lot of space and they're insect magnets, and this is area that's just not productive for you. Using the tissue culture instead of using moms, we can, in that sterile environment, we're going to clean them up, and we're also going to deal with that virus, which again can knock out a whole room of moms. If one mom gets it, and some bros are coming around and kind of massaging the mom, and then they go over to the other mom and they massage that mom, guess what? Everybody's got the virus, and you can lose a whole room. So tissue culture allows us to get rid of that virus because virus and pathogens tend to move through the vascular tissue, move through the plumbing of the plant, but the apical meristem, or the growing tip of every shoot, the vascular tissue actually starts down here. So the virus load is 0 or very low here. So if you can isolate this tissue and grow a whole new plant from that, sometimes you got a heat treat or whatnot, now you have a virus-free plant. So that's what one of the perks of tissue culture is. It allows you to extract that specific tissue that's healthy and base that whole new plant on just those few cells. Now, combination of getting rid of all those pathogens and virus and resetting the genetics, you get a happy new plant that's going to reproduce and it's going to be highly productive, more so than previous generations, and you get a happy customer that's got a highly productive plant. This is one example of same plant planted at the same time. This one has been through tissue culture. This one hasn't. You can see a significant increase in the rate of growth. And then this one's just crazy. That's a fan leaf. This is the average size of a fan leaf prior to tissue culture. Once you kind of reset those genetics, they can really pump some energy towards growth, and you get some bizarre and highly yielding plants. The other great advantage with tissue culture is it's an exponential growth curve. Every time you start a little plant, 2 weeks later it's got 4 little plants coming from it. You cut those up, you start over, you go from 4 to 8 to 16 to 32, and pretty soon, within several weeks, you can have plants up into the millions, and all of them very healthy and virus-free. So hopefully I've given you an idea of what's tissue culture, why is it there, what's it good for. It's good for, if all you're doing is cloning, you tend to accumulate pathogens and disease and epigenetic programming, i.e. it's remembering it was any stress it ever experienced, and that'll take an impact on yield. Tissue culture helps you reset, get your most vigorous plants that you would want to start with any new crop. So I'm going to talk quickly about starting material. You got some choices right now. You got seed or you got transplants. Why consider transplants or one or the other? I think, if it was up to me, seed would be awesome for everything. The issue is all that variability right now. None of these guys have been very much back-crossed. There's a lot of heterozygosity. Essentially, every seed you get is not an identical twin. It's like a brother or sister. They kind of all look the same a little bit, but you get them drunk, they become very different. You expose these guys to any sort of stress, and 2 plants can behave completely differently, even though they kind of are supposed to have similar genetics. While in a transplant that's based on a tissue culture clone, each one of these is literally identical, both in its genetics and in its state as far as its epigenetics and pathogen load. So they tend to act very uniform and predictable. We decided to go this rooted cutting route for a specific kind of market. This is some glimpses. We have partnered with Savano Nursery to do this high-scale production of rooted cuttings. So what we're interested in is helping you guys if you want to get into a kind of a hot new market. For example, CBGs, as Ben was explaining, CBG is very hot right now. Good high quality seed for CBG that's very low variability, That's going to be a couple years out. This used to take 10 years, but with molecular techniques, you can get it down to 2 to 3. But there's still a window where if you do seed, there's a high risk you're going to have crazy variability. That's what we call the Adavina zone. We can get that hot new plant, we can send it through tissue culture, we can clean it up, and for the first 2 years when the prices are super high and it's worth your time and effort to spend the money on transplants to get that super yield and that super premium, that's where we come in. And we make those high quality rooting cuttings for that zone where things are hot and you want to capitalize on a high demand market. We try, how we culture, we try to ensure everything that we do gives you a seedling that's going to produce exactly what that COA says it's going to produce. So here's some things that you want to think about, whoever you get a transplant from, you want to look at some of these various components for success. Of course, genetic potential, pest and disease, essentially the architecture of that cutting has a big effect on how it's going to do in the field, how it's hardened and acclimated, and its handling and shipping. If they ship it wrong or if it spends too much time in the dark, you could get it and it immediately goes to flower in March. That would suck. So all of these are things to think about. The first and most important is, of course, that genetic potential. What is this thing, what am I buying? What is it supposed to be able to do? The best place to start there is that COA. We've been talking about COAs a lot. Let's actually go and I want to show you how to read one if you don't already know how to read one, because they can be a lot different, but they all have certain similar characteristics. We're going to look at this one from C4, which is one of the labs that we use. One of the first things you should look for is the company name. Now that may seem obvious, but a lot of these are just purely fabricated from some dubious vendors. So you'd be surprised that when they actually pull out a COA and send it to you, it may have never actually been theirs. So I guess it depends on how crappy they are at Photoshop. But start there. Look for the particular. Are they actually giving the strain or the variety that you're interested in? One of the most powerful things you can look for is a QR code. If you take that QR code, it should take you to an online version of this, which will be your backup to tell you that this is legit or not. It should look exactly like the one presented to you. So again, sometimes these are just cut and pasted together. Here's your crop. It's always great to work with somebody that takes pictures, because sometimes what they could actually be doing is testing extract or flower or leaf or butter, who knows. So it's great to see what they actually tested. Your THC and CBD, these are just your measures here that'll give you the direct, at the time it was tested, this is percentage per dry weight, and this is the ratio. Now when you're testing, we test our crops over multiple weeks just to kind of nail down exactly when they start to go. But you can use, because THC and CBD tend to move together pretty well. Even if you test early and these are way low, let's say this is like 0.01 and 2 CBD, that ratio tends to hold pretty well, and you can use this ratio to predict once this hits 0.3, where would this likely be? So just do your ratios, your math, and that can give you a projection of where you're likely to be by the time this guy gets hot. Any questions on that? Then you have your breakdown of all the various cannabinoids. Again, some of these, just depending on the market and the time of year, can be more valuable than others. This particular one doesn't have any CBG or CBC. But again, this will give you a nice breakdown about what else is in there, and might there be a nice cocktail in there that would sell as a flower or whatnot. You're looking for no foreign matter. That's literally just, is your plant full of mites or whatnot? That would be good to make sure no. Nice little breakdown of how they calculated the CBD versus THC. They just kind of give you the ratios of one to the other. And then again, look for another QR code because there's a lot of photoshopping going on. So if these two are consistent, that tells you it's a legitimate lab that you can most likely rely on. So anybody have any questions on that, those COAs? Yeah.
The THCA and the Delta 9 THC, the 0.877 of the THCA, is that kind of the formula that everybody's using?
Yes.
Convert? Okay. So if your field just has THCA, is that what they count?
Well, depending on how the regs go, that should be what they're counting now, but if they may include additional measures. Okay, so the other aspect of genetic potential where those COAs really become important is this uniformity, and why seed in an early part of a market can be risky for you guys because there's a cutoff. There's a point in which you're screwed. So if all the plants were the same, if all of them were going to flower like in a transplant and all behaving in lockstep, you can see how here we're looking at two strains from the hemp mine and how they change in CBD versus THC concentration over time, over the season, and repeated testing. You can see that for the most part they're moving together relatively closely. But imagine if some of your plants are actually started flowering 2 to 3 weeks before others, even a small population. You can then imagine how just a few plants that are way ahead of everybody else could knock you out, make you to state regular, make you look like you're hot, even though the vast majority of your crop is actually way behind and way low. So that's where that uniformity comes in. If your crop is behaving differently all over the place, you could get real unlucky and just happen to be sampled a really hot spot just because it's further along in its life cycle, just because there's so much variability in that seed. So that's one of the big risks with lack of uniformity. Also, this shows you that even though sometimes you'll get a COA and it looks super awesome, at the total potential of maybe 20 CBD at the end of the game, but that's a super hot. That's when THC is up near 0.7. If you don't harvest by this point, you're going to be hot, and you're actually going to get around 7% CBD. So we work on everything else. We make sure that the genetic potential is well characterized. We'll give you all the COAs you need. And then our facility works on the pest and disease and health and vigor, making sure we work with Savano Nurseries down in Tucson, a very large nursery famous for their desert trees and shrubs. And we're helping them ramp up to become awesome propagators of hemp. So here you see those mothers in a highly controlled environment. We have the cutters come along in sterile conditions, carefully cutting and prepping the cuts. We have a high throughput line. We have a pretty sophisticated propagation houses where they're constantly misted and fogged and eventually hardened off to form disease-free. We don't use any sort of non-organic pesticides. So by the time you get your plants, they've been ready for the Arizona outdoor environment because they're literally in the Arizona outdoor environment, ready to thrive in your particular fields. Guaranteed pathogen-free. And here's some of the beauties right there that we're trying to dial in, making sure you have that great root zone, just enough where it's going to start sending roots out immediately, enough development so that it's enough caliber here that it can withstand any sort of environmental stresses, and a robust enough foliar that if you're doing a transplanter, it's going to make it and not fall apart. So again, everything designed to ensure transplant success. So that's it for us. I want to thank our team, and again, thank you guys for inviting us out. Yeah. Any questions, please?
I've heard people say that if you take cuttings from different parts of the plant, it will produce a genetically different cutting. Is that true?
No, it'll be genetically identical, but if certain parts of a plant can endure different stressors, and in fact, part of the problem with taking cuttings from different parts of the plant as well is the gene expression. Some of these leaves are shade leaves versus sun leaves, and some of them that programming may stick, and you'll get plants that grow up to be shade leaf plants. So you're really messing with the expression, but the genetics will be the same. That's a good question. Yeah.
I have a question about viruses, of course. So whenever you have a symptom, it's just not one virus. It's almost never just one virus. There are very few viruses that are very dramatic and show the symptoms. But to see the symptom that you are seeing in there, there has to be more than 1, 2, 10, 20, you name it. So more than 90% of the viruses, they do not show symptoms.
Right.
So you can have a mother plant that has a virus already in there, but you do not know it. So how often are you testing your mother plants for the viruses? Are you really testing them on a regular basis? Like how?
Oh, yeah. So right now we're at super high intensity, so almost every other week.
Every other week?
Yeah.
All the viruses that have been listed in all the crops, or just?
Well, the indexed ones there aren't that many high quality labs that will do cannabis, so we're working with a good California lab that several of the larger grows in California work with to all the characterized viruses. But good question. Any other?
Question. What's your opinion of root-bound transplants? Are they worth planting? Do you swarm or do you just throw them away?
No, we would never sell them. And essentially root-bound, they've woven their own basket. So what tends to happen is they almost immediately go to flower. Yeah. That's a good question. Any other questions? Yeah.
So we have very large acreages in here. When you're talking about growing in 10,000 to 1,000 acres, how realistic do you think using transplants versus seeds? In terms of labor and the money?
We'll get Ben started again. It really depends on your market, and if you have a downstream, like one thing that we will do if you choose to work with us is we're going to make sure, as Ben said, you start with the end. If you don't have somewhere to sell it before you put it in the ground, we're all screwed. So it really depends on, do you have, CBGs are going to be super hot this year. We got those ramping up right now. And we're going to help you find an extractor that's going to give you a good rate and that's going to be reliable and take your crop. And then as you work through your crop, we have a test field on our own facility. So we'll be able to anticipate any problems that you may have as a farmer as you're growing out this crop. And near the time when you need to start testing, we're not going to give you a day because that's meaningless, like harvest by then. What the hell does that mean? Your environment's going to be different, your climate's going to be different. So we're going to help you either know when to test, or we can come out there and facilitate that process so that you can time it perfectly, so you're going to get the maximum CBD without risking getting too hot and losing your crop.
Are all your facilities insect-proof?
Is there such a thing?
Yes.
They're all.
When you're getting transplants, sometimes we see some insects problem, disease problem. That is not our problem at all, and then we're just getting it from the transplant. So
All of ours are.
everything you sell should be everything in your facilities should be insect-proof.
All
That way
facilities
you're
have
not sending out
been.
insect. Especially if you're shipping it outside this area.
Oh,
where
sure.
That insect may not be a problem, and they will form a epidemic when they go to the new area.
Yeah, we had Savano had all their major houses netted with threat-proof netting. So, good question. Was this useful? Okay. Thank you very much.
So our next speaker is coming from Kyogen Seed Company, where he's going to talk about hemp genetics, the benefits of feminized seed, and day-neutral plants. So, Mr. George Workman.
Hey guys, I'm here to really run through our 2019 commercial trials and kind of get you guys familiar with what we've learned over the past couple years in cropping hemp. and discuss some strategies around planting densities, calendars, things like that, and what your tools are. We think that as a hemp farmer, it's all about strategy, and we want to help you figure out what that ideal strategy is. So a little background on Kaijin. We all come from the veg seed industry. I'm personally come from agronomy. Met my partner Christopher Hone at Cal Poly Pomona, where we actually did a lot of work with lettuce and drought-tolerant seed germination out here in Yuma. Actually did a few things back in the day. And we had an idea of, building a germplasm bank and waiting for the day that hemp and cannabis would become more familiarized with and legalized for the rest of the country. Help with the medical and all the other benefits that you all know about. Since then, worked in agriculture, growing vegetables and various other crops, and Chris went on to get his PhD in plant genetics and has led Syngenta's watermelon program. We came together 2017, gathered our germplasm, put it together, and created a strategy to model a typical vegetable seed company, and really trying to use the 4 key principles to get farmers to success. And we think that's not really going halfway there, but I'll start with the first 2, I guess. The seed industry experience I touched on, but we think the technical support is really what's lacking in the seed side of things. And when people say snake oil or shifty, shady seed companies, we think a lot of that has to do with lack of knowledge. We think a lot of those people, we're not sure how, maybe half of them are actually trying to commit a devious act, the other half just don't know what they're doing. So we think that the technical support is extremely important for farmers trying to get into a space that has a lot of risk and a lot of, especially upfront financial risk associated with it. The third thing is the quality control piece. But we think quality control is essential. And most of the time when folks make seed, they'll go around, they'll harvest the seed and sell it. We think it's extremely important to do a genetic testing on the seed and make sure there's no males in every lot, segregate all the production lots into different batches, and make sure you properly evaluate each batch. And not only that, but to do commercial trials with growers, universities, like with U of A, Bindu. She did a wonderful job at running a winter trial, which we've been learning quite a bit about growing in the winter in the desert, and I'll touch on that later when we go through planting calendars. But as far as quality control goes, we think that's one of the 2 biggest lacking things in this industry is customer service and quality control coming from the plant providers, seed producers, and breeders. Yeah, and our whole thing is really we want to focus on agronomy. We don't look at like farmers as our customers, we look at them more as partners. We're like evolving in this industry together, and we want to learn together. So the more we can trial, the more we can share data, the more we can advance this thing and lower each other's risks as business to business operators. So I'm going to start to talk about, can you just scroll actually? Because I don't think this thing's working out for me. Just scroll up to the top, to that top half of that slide right there. Yeah. So we just really want to get people familiarized with the anatomy of the hemp plant and what your options are with seeds. So you have dioecious seeds, which are male and female. I think you guys learned a little bit about that earlier. And you have feminized seeds, which can range in quality depending on who's producing them, providing them. So our whole promotion is the feminized seed, primarily because with dioecious seed, you're losing 50% right off the bat. And as the price of hemp drops, your net yield per acre is extremely important. Your inputs are expensive, your fertilizer, in some areas, land, things like that. So we're always looking to work with the farmers on understanding that net yield and that bottom line and trying to fix that. The thing about male plants is obviously it's really hard to get them all. When you're dealing with a dioecious crop, say you have 2,000 plants, 1,000 of those are going to be male. In a feminized crop, if you have 2,000 plants, 1 of those will be male. So you can imagine the amount of effort and input you have to put into to go and find all those. And then even more of a challenge, they show up at different times. So you'll have early showing males, late showing males. And if you miss one true male, that's actually pretty devastating because it can hit a 5-acre block and completely degrade the quality of product. With the feminized male, so the 1 in 2,000 in a quality feminized crop, it still will do damage if it's missed, if you don't find it, but the amount of damage it'll do will be much smaller, much more controlled, because again, the 1 in 2,000 I'm referring to in a properly feminized crop is not a true male. It's still a feminized induced male, so it's producing that pollen that's much less vigorous than a true male would produce. And first and foremost, be a good neighbor. If your neighbor is putting a lot of effort and input into their field, they really have a market that is asking for a seed-free crop. And as quality increases, that's all good. You can say it right there. No, back up. Yeah, right there's fine. Yeah, and so if somebody's really putting in that input and you're down the road and you don't quite have the input capital to start up and get involved with hemp, you're kind of cutting the corner, could in fact impact your neighbor's operation quite a bit. So those are the reasons why we promote feminized seed, and we think it's gotten quite a bad rap because folks have not produced it properly and they have not had the proper quality control to prove it. So what we do is we do commercial trials. We prove what our germ is. We prove what our fem is. And through, rather than talk, which I could do all day, it'd be better for someone to come look at it. And that's how ag works. We operate in trials because there's no bullshit there. So up to the top a little bit. So the next thing I'm going to talk about is day length dependent and day length neutral varieties. Day length dependent, they really need the days to be short to flower. So primarily generating from regions closer to the equator, so forth. So when the fall time comes and the days get shorter, flower induction occurs and everybody's kind of harvesting their crops right around September, October, November, and that's when the majority of the country harvests their hemp. And these are the types of crops that most people are familiar with, or the types of genetics that most people are familiar with, like the cherry wines and things like that. We also think that day neutral varieties are a very good tool for farmers to use as well. And then those are also known as autoflower. And those come from regions of the world like Russia, where the days are very long and the plants have had to evolve over time to figure out how to flower and reproduce. And so therefore you can be harvesting a day neutral variety in June, July. And I highly suggest any of you that are interested in market rates and how they fluctuate in hemp throughout the year to check out PanX Exchange. It's a third-party provider that talks about spot pricing in hemp, and basically it shows the fluctuations in spot prices within hemp throughout the year. And you can see there's an obvious incline in price in June, July, August, and then the price just tanks when all these day length dependent varieties come off in the fall, because you got the Midwest guys combining, you got everybody going. So we feel that they're both good. That we feel like they're both a great way to spread your risk, and depending on where you are and what your goal is, there's an option for both. If you're transplanting, if you're not really keen on direct seeding, we always recommend the day length dependent, because the day length neutral has much more of a sensitivity to root stress, things like that. So we always recommend direct seeding that. But there are plug options that are available as well, little net pots and things like that. But we've found that the highest quality plants come from direct seeding. And it's obviously the lowest cost to the farmer to run it in the ground that way. The beautiful thing about the day length neutral varieties, in our opinion, is the succession harvest. And you can do this to a degree with the day length dependent as well, but the day length neutral really are good for block plantings. So when USDA compliance is coming online and things like that, you want to really spread out your compliance tests. So for example, if you have a 100-acre field and you split it up 5 ways, 20, 20, and so on, you have all of a sudden, instead of 1 compliance test on 100 acres, you have 5 compliance tests, all planted differently 2 weeks apart. So instead of having to harvest your whole 100 acres in 2 weeks, like the USDA is recommending, you can now harvest 20 acres in 2 weeks and then do it again in 20 weeks. So very similar to whatever, broccoli or lettuce, where you guys are planting. This is pretty much a similar concept. But once you get closer to the fall and the days are hot, we really recommend the day length dependent, and that's primarily because these day length neutral varieties, they originate from such northern climates that the stress of the heat in that juvenile stage of their growth is actually very detrimental. And so they like to be started in the spring and they like to finish in the warmth. And so as you're and I'm gonna go through a planting calendar here, and this will all make a lot more sense to you when I run through our slotting calendar. We can show you how you can start in the spring and finish in the fall and have a nice cascading harvest rather than this big wave that comes and makes you freak out at the end of the year. And we can move on to the next slide. So I'm gonna just run through a few photos and just discuss things as we go. We'll go to the next one. Yeah, next slide. So we do everything in high plant populations and high planting densities, and we've actually found that by increasing plant count, you're able to increase your bottom line per acre considerably, and not leaving so much space in between your plants. I don't know, I'm sure you guys are pretty familiar with the bush types, a bunch of soil in the ground, blank spots everywhere. This type of operation here, that's 19,600 plants per acre. And so of course you're thinking, oh my God, the seed guy's trying to get me, huh? Right? But no, we're actually increased our volumes of seeds and are trying to realize an adequate price per acre for the farmer, and really trying to work with the farmer. As commodity prices drop, we want to figure out what that ground is. So again, we're not working with the client, we're trying to work with the farming partner in that sense. And so what we're averaging here with these increased, everybody's used to kind of that 2,000 pounds per acre, but once you're really doubling, tripling, even quadrupling those densities, you can get it up to, we've seen it as high as 4,000 to 6,000 pounds an acre of flower off the stem. Yeah, and we'll move on to the next one. So this is a berry blossom crop. And this is a full term, and it's planted a lot later. So these plants are actually waist high. They're not like these huge 8-foot tall plants that are gonna break your combine or break your workers' backs. They don't need a saw to cut them down. And you're left with a great harvest index. And so harvest index is basically the desirable portion of the plant is higher in ratio to the undesirable portions, such as like the stem or the leaf. So when you grow a shorter plant like this, you can see the majority of that field is buds. It's not a bunch of stems and bushes and everything else growing everywhere. So another reason why we think that you don't need to plant a big tree orchard style type deal in May, June, because you're going to grow a lot of stem. And if we're in the fiber industry, that's great. Buy a fiber variety. Don't buy a CBD variety if you're going to go that route. And we're growing buds. So next slide. And then, so this is a picture of a berry blossom flower. And I kind of wanted to talk to THC compliance on this one, because this flower right here is at 0.4 THC. So it's hot, and it's I think 11.5% CBD. So if a farmer were to be harvesting this last year, it was totally compliant, no problem, because it's Delta 9 plus THCA gives you total THC in California. I don't know what the deal is in Arizona, but I think it's a lot more strict. I think you have a 2-week, but it's only Delta 9. But in this future USDA regs, you have 2 weeks to harvest, and it's a total THC of 0.3. And so instead of waiting until this peak maturity, you need to be harvesting 2 weeks early or a week early. And so it's really up to that farmer to really monitor those rates as they go. And it's like with the $50 test twice a week, we think you can actually stay on top of it. And it's really all about the farmer staying on top of it, and we're here on the technical side to help them do that. We have an in-house lab where we do all our breeding testing, and we're thinking of expanding that to meet R&D sampling needs for farmers to make sure that they can kind of keep up with stuff. Little background on our breeding efforts. We have 180 populations, and we focus not solely on seed production and just generating cash flow, but we have a really long range vision in this thing. we see hemp being grown from seed, very similar, or hybrid seed, very similar to vegetables. And there's a lot of comparisons. I don't know if you guys are familiar with the Veggie Days, where there was open pollinated veggies. You got 5 varieties. They're all the same, but they all have different names, and they're all supposedly so much better than the other variety. This is the same thing. We're dealing with all that all over again, and everything's kind of shifting from this whole open pollinated shifty deal to hybridized seed. And so just as the years go on, scaled breeding populations, real PhD breeders using genetic tools, high quality sequence genomic work, understanding cannabis pathways, those are the types of things that are going to get this crop modernized and efficient and scaled for a farmer. And it's not going to be a dollar seed. It's not going to be a 50-cent seed. It's going to be a comparable to a vegetable crop here. And we'll go on to the next one. So this is one of our varieties called Maverick, and it's a auto-flowering variety. Again, gets waist high. So when you're looking at the harvest index, instead of having a bunch of stem and 1,700 plants per acre, you're doing about 17,000 plants per acre. And then that adds up. All those buds are actually yielding a higher net yield CBD. And this year we're working with several processors, and we're actually going to be able to pinpoint liters per acre, and that's how we're going to be advertising these varieties in the future in terms of oil. But we feel if you're shooting for the flower market that you shouldn't, we saw everybody lose their ass this year on this flower thing. And the reason is because they went and saw $200 a pound. I'm going to harvest everything and try to get $200 a pound. And you know what? That killed them, because some of them put in like 120, 150, maybe even $300 a pound to get to that $200 a pound. And the quality wasn't even there to meet the spec of those buyers. So then they were sitting on this product that all of a sudden would have initially cost them, $10 to grow, that now cost them $150, and they can't even sell it because it's not hitting the spec. And that's another thing this industry really needs to understand is the specs. And if you're going into this market, you really want to understand the specs. Even if it's a prospective buyer and you're growing on the open market, understand what particle size do you want your biomass. Understand, do you want it homogenized? Understand what kind of flowers, what kind of flower demand, what kind of terpenes, what size flowers, all these types of things matter. So what we think the best way to go, this is again just my opinion here, is to really come through your crop and selectively harvest the most premium portions of that crop, whatever percentage that is, but it has to meet the most, like the top 5%, 10% of your fields should maybe go to that flower side, because that's your premium, and you'll actually be able to, it'll actually be worth that input you're putting into it, and it'll actually be worth it for the buyer to be purchasing it at that rate. And you'll actually probably be able to sell it versus trying to take all those bottom branches and try to just milk the whole field for everything it's got. So once you take those portions out for the flower, then the rest of the plant can either be dried in the field and combined or harvested mechanically into where, I've seen those tobacco cutters where it cuts the bottom of the plant, throws it in the back of the truck, and then you can dry it in a facility or a commercial dryer. But we feel with the way that the prices are going, and the way that this industry going, is it's going to continue to be lucrative, but it's going to be the efficient, mechanized farmers that have a good strategy in place that are going to be the ones that win. So I'll be the next one. Just some examples of that autoflower bud. It can still range as big as those, you see these big 7-foot tall plants, and they have those buds on them. That's a similar sized bud right there on a waist-high plant. So just a little perspective there. Next one. And we did, and this just shows the genetic variability that exists in seed right now. Very similar to the open pollinated days, where you'll have some plants that are taller, some plants that are shorter, but at the end of the day, as long as your compliance is on point with that top 2 inches of the plant and the 5 plants being homogenized, it's all about your net yield per acre. So you can cherry pick your best colas out of there and still combine harvest the rest and still get a great yield out of it. And if you scroll all the way back up to the top, actually, we'll show you our other variety that has 90% homozygosity, and it's basically almost to hybrid form, so it's very uniform, finishes all at the same time, but then again also has a lower CBD content per plant. So this is Pipeline. The last field we showed you with the genetic variation was Maverick, and the CBD ranges in Maverick hit anywhere from that 11 to 15%, and it's one of the few day neutral autoflower varieties that can actually get that high. In fact, we haven't seen any that get that high, but I'm sure they're out there. This right here is Pipeline, and it hits 8 to 10% CBD, and it's a lot more safe on the compliance side, and it's very uniform, but a lower CBD producer and a little more sensitive of a plant to grow with less vigor than the Maverick variety, which has the genetic variability, the multiple phenotypes. But yeah, we feel like pictures say 1,000 words, and when people say that there's the feminized seed never gets that high of a fem rate, our fem rates have been hitting 1 in 4,000, 1 in 8,000 consistently, and that's just to proper practice and proper quality control. And I think you could see that in the photos when you see a full field. If it's obviously not 50% male, right? Those are all feminized seed. This is a Maverick flower right here. So this is an example of what I would call that 10% of that field that you would harvest for that premium price point for flower. Like you go out there, you grab all these tops, and then anything that's subpar, blemishes, whatever underneath it, that are small, harder to trim, have more leaf matter to it. That's all stuff that should just be going right through the combine and turned into oil. Keep going on those. So we provide tech sheets for all our varieties, similar to any vegetable you guys grow. Just kind of showing the COA at the bottom here, but we provide multiple COAs. We provide like a stack of 5 or more, just showing the range at different times of the week. And if you're buying products or plants or anything from somebody, I suggest you don't just get one COA. I suggest you get multiple. And so you can get an idea of what this crop does whether it's at a different time point of harvest or whether it's just the phenotypic variation. The more you know, the better. And so we have like a little bit of terpene profile for the folks that are interested in flower. The days to maturity. So this is 85 to 90 day variety versus like a cherry wine, like pick right here, which is a full term. That's just depending on when you plant it. It has like a 9-week flowering time from when flower induction begins. Again, that's also dependent on latitude, which is why we're so keen on trialing based on latitudes. We started up in Washington and Spokane, and then went all the way down here to the imperial border and tested different densities, different time slots, and developed these planting calendars. And if you want to just scroll through the next couple of these little deals. So this is our variety slotting calendar for coastal California. And in the yellow, it's very area dependent, and of course if you have hoops or not. And we also show the ideal slots for planting. And so the harvesting of these varieties isn't shown here. You'd be harvesting actually in the red in these sections here. And so your goal, if you're harvesting in July and August, that Panex exchange, that spot pricing thing I was telling you about, will show you that the price points for your hemp are going to be up to 4x or 2x to 4x of what they are in December and January. And not only that, but you're not competing for an extremely extreme quality standard or price point. Like your product's actually in demand at that time, and people are hungry to buy it because the market isn't saturated. And if you keep going a little more, I think there's 2. Skip this one, and we'll go to the desert. Keep going. And the southern desert's right here. And so we found this year, we learned a lot growing in the desert this year. We did some trialing, some folks bought seed and did commercial grows, and we learned quite a bit about what happens to CBD levels and what happens when plants don't get light. And we also learned what happened last year when people planted in the peak heat of the desert, and it didn't work out too well either. So it seems to us, from what we've learned, the successes of the folks that were successful ended up running full season varieties in the fall. That's the berry blossom that we have to offer. Like these 3 sections are 3 different varieties. The yellow is basically area dependent, like if you have a microclimate that's favorable, and the green is suggested. So we feel like the full seasons, like for these cherry wines, if you're planting them in the fall, they work really great. The days are short, and everybody thought they were going to flower after planting them. I mean, maybe a transplant we think would flower, but if you were running a seed plant and you're planting in the fall in the desert, you're actually going to be able to get a good, decent sized CBD content out of it. And instead of getting a big bush, you're going to get a day neutral auto flower style plant out of that full season variety. Versus the day neutral varieties, we suggest planting here at this time. Primarily because folks that planted full seasons or past January, anytime you're planting a full season in February, March, or April, that thing is going to start to revegetate in these summer months, and it's going to turn into a 20-foot tall plant. I don't know if any of you guys saw that deal in Bakersfield where the guy grew, like 30-foot tall plants, and it was just out of control. That's pretty much what happens. They need a 12-12 life time point to flower, and that 12-12 time point needs to be prolonged for the entire duration of that 9 weeks. So if you're, say, planting a full season in March or April in the desert, it'll start to flower pretty small, then it'll revegetate again, and you're still going to end up harvesting September, October, November. You're just going to have this whole drawn out deal. Doesn't make sense. So, yeah.
Why would that be bad? Don't you want a really big plant with lots of...
Well, that's why I'm coming back to the whole harvest index thing. If you're growing fiber, absolutely. You're getting majority stem. We're trying to grow buds. And the longer you're in the ground, as any farmer knows, the more risk you have, the more susceptibility to risk. The bigger your plants get, and if so, the lower densities you have, the more space and dirt and all this other stuff you got going on in between plant to plant, all that's weeding, all that's management. You have crews out there. You're putting inputs out there, fertilizer, water, you're leasing your land. You want to keep that land as productive as possible. So constantly turning and burning crops is what it's all about. And I think if you go to the next one, I don't know if I have much more, but yeah, that's just our deal. Is we really try to increase our seed inventories to factor in practical costs per acre for the farmer, so they can not sacrifice on the right planting densities. I think a lot of folks have been doing the whole 1,700 to 2,500 plants per acre, but we've found that even on full season varieties, once the cost of feminized seed can go down, you can get really high up there in plant count, as long as you're in an area that doesn't mold. Like for example, if you're in a microclimate like Watsonville or something like that, or if you're out in Salinas and you're getting fogged in, yeah, you want to put some space in between your plants. But if you're in a dry region and if you're pretty comfortable with your humidity, you can go pretty dense with hemp. And again, that's also genetic specific. So you can have varieties that mold really hard and varieties that don't mold as hard. So knowing your microclimate, knowing your starting genetics, knowing all these things, that's what it's all about. So just the more you know. So that's it. Thanks. Any questions? Cool. Yeah.
What region was the southwestern desert testing?
It was the Imperial Valley. Mm-hmm. Yeah. And one was in Winter Haven and one was in Brawley. And the CBD percentages of the day neutral varieties that were planted in like November and December, they were as low as like 2 to 3%, really low CBD percentages. Because at the end of the day, these things need light and photosynthesis, and they need energy to produce these phytochemicals, and they need it to produce the buds and the flowers as well. So a lot of plants had like nice green field. A lot of folks had nice green fields, and everybody there's a lot of just confidence the whole way through up until the very end. But at the end of the day, they were growing like a lettuce. It's like a vegetative deal. you're growing phytochemicals, you're growing buds and flowers, you need energy. The plant needs energy. So, yeah. Is that all?
Is there a benefit of growing multiple varieties to hedge your bets?
Absolutely. That's one of our big things is we think in anything that's high risk, you should avert your risk. Plant in different times of the year, plant different varieties, and even when you're planting your full seasons, people are like, oh, you gotta get all your full terms in before June 15th or you're done. That's not the case. You can block plant your full terms even. So there's a lot more to it. But in the desert, I would strongly recommend everybody really watch out for those extreme conditions, extreme heat. I'd extremely watch out for you. But I think you in the desert, the most money-making slot in the desert, personally, I think, is planting in February, March, harvesting before anybody else harvests the day neutrals. That's the earliest anybody in the United States is going to pull a hemp crop, and that's where it's going to be at the highest peak demand and the highest price point. Yeah. Cool. And if you guys have any questions, you can just go on to www.kyogene.com and you can send us a little message on there and I'll reach out to you directly. Thank you.